fluid around the tumor environment, also called ascites. And there are a lot of papers that have been presented on that from a while ago. So once you go through this first step and you identify your biomarker candidates by looking at the changes in some biomarkers or proteins between these samples, then you can go into the verification stage where now you increase the number of samples and decrease the number of analytes that you're able to simultaneously detect. And you can also move this from the discovery sample matrices to blood in a larger population of people. And so blood's a little bit easier to get than tissue, or proximal, a lot easier to get than tissue and proximal fluids. And then you can begin to see if these changes that are being observed are significant and relevant, and if you're able to detect them with high precision and reproducibility. And then finally, what we're all sort of striving for here is application of these methods to clinical validation, which means, can a doctor look at the results and may make a diagnosis or use it in a diagnostic way for treatment? And this is an area that LC-MS has not really flourished in yet. There are a few methods that are used in the clinic, but we're not here on a very large scale yet. And there are a lot of factors that affect that. And so some of this is what I'll be going over today. So how does discovery-driven proteomics different from hypothesis-driven proteomics? Well, here, hypothesis-driven is you have an idea based on some information of how much is changing in a particular target. And so you follow that target through different samples, different environments, and you see how it's changing, the extent and the reproducibility, the biological variation of that. And then ideally, when you start with your biomarker candidates, you want to make sure, is it found in blood? Is it higher in cancer? It's easier to detect a biomarker increase than it is to detect a decrease in samples. And so this is why we always usually try to look for something that is increasing in our samples, and then determine if these biomarkers are worth evaluating. So, whoops, moving on. How do we do, well, what's the difference actually between quantitative mass spectrometry and quantitative proteomics? So quantitative proteomics is when you're detecting the changes in as many peptides in a sample as possible, and then rolling them up and applying them to changes in the proteins that they come from. And so you're not really narrowing down your list, you're trying to look at everything. So you can imagine just if you're out in a field of flowers and you're looking at all the flowers and seeing which ones are taller and which ones are smaller. How does that compare to quantitative MS or targeted? In this case, you'd be looking at those field of flowers and you're like, just go look at the yellow ones. I'm going to ignore everything else. They're still there, but your focus is on a smaller subset of the peptide population. So the idea here is you're asking the question, how much of peptide X is in my sample? And then you can quantify it relative to a standard. We often use isotopically labeled peptide standards, which can be synthesized and purified when you know the amount you're spiking into the sample, and you look at the peak area difference or the difference in the signal between that internal standard and the analyte. The beauty here is the C13 and 15 labeled peptide elutes in the same place and behaves very similarly to the analyte, but it just has a different peak area based on what you're spiking in. And another option is, so that would be considered like a labeled analysis. If you're doing label free, you might have two different disease states or a control and a disease state. And now you're looking at that same peptide signal in two different samples that have been acquired differently in time, and you try to analyze those and see what the change in the signal would be. So this is relative quantitation or label free. So how do we do quantitative proteomics? There are a lot of ways we can do this. The labeling strategies are a little more expensive and intensive and could be limiting. So we've got metabolic labeling like Cylac, chemical labeling where we go in and chemically derivatize some kind of entity in the peptide or protein sequence. Label free is when we do not use labels and we have to analyze our samples one at a time, which is the expense here is often in the amount of time it takes to generate the data due to the need for additional replicates, or targeted proteomics. And so designing methods specifically to look at just a few smaller number of things than many, many thousands. So going into each of these individually to give you a background on what they entail. So stable isotope labeling with amino acids in cell culture, it has the acronym Cylac. This was originally presented on many years ago, and it's meant for use with growing cells in culture. So if you have a cell line and you can treat it with a drug, or you've got something that's resistant to a treatment, you can look at how the protein expressions are changing between these two cell states, and you feed them heavy media. So you give them heavy or labeled amino acids in cell culture, and they begin to incorporate those amino acids in their protein sequence. And so usually it is lysine or arginine that are incorporated in the media, and then those heavy amino acids are now included in the protein sequence. And when they're digested, they are also in the peptide sequence. And so here we've got the normal amino acids on the left, which would be light, and then media labeled with heavy amino acids, which would have the asterisk. And over time, so once these cells are labeled and you go to harvest the cells, you can combine them immediately. And so any sample prep steps that these cells go through, they are seen equally and will have the same level of variability. And this is one of the most ideal situations because you can combine your samples very early on in the process. And what you see from a mass spectral standpoint, or from the mass data, is you'll see doublets. So there will be the light peptide and the heavy peptide that's a certain mass to charge away, and you can quantify relative changes in these peak areas based on experimental parameters. So in order to do this, you need metabolic labeling reagents like mammalian cells. This is not like classical pulse chase experiments with radioactive isotopes, so perhaps a bit safer. And then the idea is you add these things into your media and you grow the cells. Then it can be done in a two state or a 3-state, depending on how many heavy amino acids you have access to. And this can all be controlled by budget as well as samples. But the idea is once you've grown all of your samples, you combine in that very first step, and then you do the reduction, alkylation, digestion, and then quantify by LCMS. And this is the kind of information you get where you'll see a light and heavy doublet in the 2 samples. So this represents the same exact peptide in pretty much the same exact abundance. And then for a 3-sample state, you've got the light and sort of the medium and the heavy in the mass spectrum. And you can pull this information out using software like Skyline to quantify the peak area underneath the chromatographic peaks. And so this quickly goes over the advantages and disadvantages of Cylac, where we have advantages, or the label is incorporated very far upstream and the variability is accounted for through this. The samples are combined early on, and the quantitation of most peptides is possible as long as you see the presence of the light and the heavy, or 2 out of the 3 if you're doing a 3-way. Unfortunately, this only works the way it's been designed with cell culture, and the media may be expensive, and only 2 or 3 samples can be compared at a time. And it is so technically this is incorrect. Human samples could be possible if you could feed people isotopically labeled food or water. And there was a study done some time ago with mice where they were fed heavy chow. But the process for this is a bit arduous, and the chow is expensive, and mice are not clean eaters. So they'll go in and the food will go around. So it's probably not something that people would want to do routinely. So another approach is using chemical derivatization, and this started many years ago with isotope-coded affinity tags, where you've got a thiol reactive group which reacts with your protein or peptides, usually the cysteine residue, and this can be modified to react with a number of different peptide reactive groups or protein reactive groups, like the N-terminus or primary amines. And then there's a linker region that you can incorporate heavy isotope like carbon-13 or nitrogen-15, or in some cases deuterium. This was very early on. And then the last portion is an affinity tag. So you can preferentially enrich anything that has been modified with this thiol reactive group. So the ICAT was used for targeting cysteine-containing peptides in digests, which not all proteins have. And the proteins that do have them usually don't have a ton. So you're getting some kind of subset of your entire proteome, and you're able to quantify changes on that level based on heavy versus light linker regions. So this has evolved over the years into iTrack and TMT, which is an isobaric labeling strategy. So on the top here, you would see a doublet, much like Cylac in the MS1 signal. You would see light and heavy. Down here at the bottom, iTrack and TMT differ because that once a peptide is labeled from across your samples, it'll have the same intact mass. And so the fragmentation is what allows quantification in the MS2 space. And how this is done is all of the versions of this light and heavy and peptide that's been labeled by these tags, if it has the same sequence, but a variety of different tags, the entire mass of that tag is the same. So these peptides from a bunch of different samples will all co-elute with different mass to charge values. With the same mass to charge value at the same retention time. And then when the mass spec isolates it and fragments it, this reporter ion region happens very low in the mass to charge space, and it'll allow you to quantify changes. Each sample, each peptide from a different sample will have a different mass fragment here. And this mass has been extended now to like 130. It's much, much higher. So you can multiplex in a bunch of different ways. But this is how this looks, where we've got the isobaric chemical labeling. In this case, we have 4 samples, and they are digested, labeled, put over they're digested, labeled, put over a strong cation exchange to remove the reagents, C18 separation. And here you see the fragment ions in this region. And then from the bottom, you can see the MS2 getting you the peptide ID. And then you're able to sort of look at these peak areas and quantify the amount of peptide that's in the sample from each of these starting sample points. Using iTrack or TMT, again, as I mentioned, the precursor ion for the MS1 scan is the same for the peptides coming from all these different samples that have been labeled differently. And when the fragmentation occurs, this is just a blow up of the previous screen. You are able to identify the sequence of the peptide. So hopefully you could go in and then backtrack it to a protein of interest. And then this very small region, in this case, it's iTrack, is looking at the intensity of that fragment ion coming from each tag. So this would, the 114 is sample 1, 115 is sample 2, and 3 and 4. And you can see in this case, we would say, oh, the 116 or sample 3 has the most of this peptide in it compared to the other 3 samples. And then you're able to pull out the information from the peak areas for relative quantitation. Over the years, the number of samples that can be analyzed at the same time increases by modifying the tag itself, the reagent, and incorporating different isotopically labeled atoms to make the mass change different in this reporter ion region. So here, this is iTrack. We're looking at mass to charge of 113 up to 121. So this is an 8-plex that allows you to analyze 8 samples simultaneously in a single LCMS run. I think now we're up to like 20 or 21, and this requires high MS resolution because some of the mass differences in the tags are actually 0.1 mass to charge units. So you need to make sure these peaks are super narrow, highly resolved from one another in MS space, which usually requires high resolving power and an Orbitrap mass analyzer. So this is an example.
Couple questions in the chat. First question, Cylac is not feasible with complex tissue lysates, let's say mice tissue lysate. Why can't you use Cylac for mice tissue lysate?
Okay, so Cylac requires the building and transcription of proteins while it's in the media. And it's usually, once you have tissue, it's usually a piece of tissue. And the biological activities going on there are going to be a lot different than if that tissue is like still attached to the living entity. So incorporation of those heavy amino acids into the protein sequence is not possible in a living being unless you're feeding them. So once tissue is extracted, whatever biological processes are going on would be difficult to relate to how it would actually be occurring in the living intact sample.
That's like kind of a follow-up. Isn't there a Cylac mouse available?
There may be these days. Actually, I haven't gone shopping. I don't tend to do the global proteomics in a routine basis. But yeah, I think anything could be available these days if you need it to be.
Yeah. And I know you had mentioned in the lecture, it's not Cylac because it's not cell culture, but Cylam in mammalian, like mice. You can buy heavy chow, and that effectively will label your mice, but the heavy chow is like super expensive. So if you ever want to like ask some of the gray hairs at ASMS, they will tell you, regale you with stories of picking heavy chow out of the mouse like litter and poop to recycle it because it's so expensive. And then we have a question on TMT. Is the advantage to TMT saving on runtime? Is that why you combine all the samples to save on runtime? Are there other advantages as well?
Yeah, so one of the major ones is exactly that. When you look at a TMT experiment versus label-free experiment, a TMT 10-plex allows you to look at 9 samples plus a control simultaneously in one run. You would probably want to analyze that in triplicate. Versus if you were to do those same 9 samples plus control in label-free, you would need to do each one of those runs in triplicate in order to get the technical variability of those measurements. And so there is like a balance. The TMT reagents are expensive if you're doing like a 1 versus 1 experiment, because you can achieve those by doing label-free by just the cost of the LCMS analysis. Whereas the reagents are expensive, or could be in the thousands of dollars. So if you buy a kit and you're analyzing 10 samples, now, yes, you are saving on that runtime by combining all your samples together. So there is a portion of like experimental design consideration based on your budget and then your LCMS availability. And when I was running a core facility, I would go through these calculations with people being like, oh, you only have 2 samples. It's easier to do label-free. And you'll get similar information, at least answering the questions that you want. Whereas if someone had 8 to 10 samples, then it would be cost effective to buy the kit and then just run the whole set in triplicate. But yes, that is a very good point. Thank you. Okay. So as I mentioned, when you get to higher plex levels, meaning more labeling reagents, now you can see the differences that you see in this labeling profile or the barcode are so close that if you don't have accurate, high enough mass resolving power, they'll come out as a single peak. So this is why using instruments that have a certain MS resolution is important to take into consideration. So time of flights and Orbitraps are the 2 most high resolving mass analyzers. But unlike Orbitraps, time of flights usually have an upper limit and you can't really adjust what the resolution is like you can on the Orbitrap. And so often TOFs are limited to 10 plexes or lower. Okay, and then for the instruments that can go to higher resolving power, you can see now, I think up here, this was showing 4 pairs of reporter ions that are close in M/Z, but now Thermo has developed these TMT kits where it's a lot more. And so you're able to, again, multiplex more samples all at the same time. And the importance of this is being able to measure your samples enough times to determine their biological variability and the technical variability in the measurements. And this allows you to tease apart what's being changed due to biology versus what's being changed due to just measurement precision. So for academic labs that don't have a lot of money and are looking for any ways to do some kind of quantitation, there are other less commonly used. So using heavy water is one where you can digest your sample in heavy water, and this O18 atom is incorporated into the peptide during the trypsin digestion. Funnily enough, funnily, I don't know if that's a word, sorry. You can actually add O18 water to your sample after it's been digested and you add trypsin again, and it goes in and it'll still do that replacement shown in this chemical reaction here. So you can generate an O18 labeled standard and like have it. You can take also your same sample that was digested in trypsin, redigested in trypsin with O18 in one sample and regular water in the other, and you should be able to differentiate them based on this heavy oxygen that's incorporated into every peptide. So not so great because the mass to charge difference is only, or the mass difference is only 2. So usually it'll be a mass to charge difference of 1 if it's doubly charged. Another option is dimethylation. Again, not superly common, but if you're desperate, these are some things that can be used, and it's done post-digestion, reacts with the N-terminus of the peptides, and it'll impart a light and a heavy version. These are deuterated, which as long as you're not going above a certain number of deuterium, like 4 to 6, they'll still have the same chromatographic properties as the non-deuterated reagent. So this just goes over some advantages and disadvantages of the chemical labeling, where advantages are we can quantify in the same LCMS run when you're doing chemical labeling because their mass to charge is different, so you can combine them before separating by LCMS. This can be done on human samples like plasma, tissue, animal tissue, and so on. Usually there are kits available depending on the goals of your experiment. There are also these quote unquote, cost-effective alternatives that can be more laborious just for that. And multiplexing is available. So disadvantages are that the label is usually incorporated post-digest. So there are a lot of variability imparted in the steps leading up to that. They're usually not combined until far into the workflow. The kits can be expensive if you get the good isotopically labeled ones. Labeling from anything may not be 100%, so that's something to keep into mind. Data analysis software is necessary, and peptide IDs are not guaranteed. So often you can get quantification of peptides from iTrack and so on, but you might not know what their precise peptide sequence is, and therefore how it dials back up to the protein level. Okay, so the last more.
Oh, yeah. Sorry. Couple questions in the chat regarding labeling. So a question about iTrack. What is the point of making isobars? Why do isobars elute at the same retention time?
So the purpose there is, I don't know, here, this is an example of the MS scan for the precursor ion. This is not just one peptide. This is 1, 2, 3. Peptide, the same peptide sequence from 4 different samples that were combined with that chemical labeling reagent. And in order for us to figure out what this peptide is, it needs to be isolated and fragmented. So in this case, you want all of the different samples with that same peptide to be isolated simultaneously. So they have these tags that have, the total same mass value. They've got the reporter ions, which differentiate by mass to charge. And then you have the balance, which helps you to total up for each reactive reagent the same total, so that they will elute in the same place and have the same mass to charge. Then when you isolate them and fragment them, the only things you're going to see are what's coming out of the peptide sequence. So you can ID it, and then this reporter mass region. And so this is where we've isolated this 791, whatever peptide this is, with the 4 different labeling reagents on it. And then we can see all of the Y product ions and some of the B, so we can identify it in the MS2. But this reporter region in chart C that's blown up, this is how we quantify from each of those 4 samples how much of that peptide is present. And so we can do it on an 8-plex. Presumably one of these channels might be a control, so you could normalize all of these signals to a control channel. And so that hopefully it's the idea is these intensities give you a quantitative measurement of these peptides in the complex samples. Does that answer it? Is that clear?
Got a follow-up question on TMT. Is fractionation always necessary for TMT combined samples?
So fractionation, which I haven't gotten into yet, that is something that allows you to take your one sample and turn it into a lot of other samples. And so some people will fractionate to get, it's so what the idea is, what it does is you take your sample, let's just say for argument's sake, you split it into 8 fractions. And by doing this, you're separating your peptides on some orthogonal means. So you're not necessarily using acidic reverse phase, which is what we use for LCMS. You might be using basic reverse phase, so high pH, which separates the peptides in different orders. Or you might be using strong cation exchange, which separates the peptides based on their cationic properties, or or I think those are the most commonly used ones, but those are the phases that people will use. And then they'll collect different fractions along the chromatographic timeline. And then each of those fractions will be then analyzed by C18 acidic C18 reverse phase LCMS. The idea there is you're taking a portion of your sample, your whole sample, and separating it from the rest of your sample, and then analyzing it individually over a reverse phase gradient. And the idea here is you are able to detect less abundant species. And so you're improving your dynamic range of peptides that can be detected from your sample. The problem is reproducibility of that fractionation and quantifying some of the signal if it's been split into a couple different fractions. And so it depends on your experimental goals. If you don't fractionate and you detect all the peptides and proteins that you may have a hypothesis for, awesome, then you don't have to fractionate. Some people will fractionate into 8 or 12 or 96 and then maybe back off the fractionation level once they figure out what they need to see in their sample or make biological relevance of it.
So many questions coming in now. Okay. Maybe this one's a quick question. For disulfide or modified cysteine residues, can you still label those?
No. Usually the interaction of not disulfide. So if a cysteine is cross-linked with another cysteine, you usually can reduce those and making free thiols, which then should be reactive with other things. But if cysteine already has been modified or like methylated, so I'm not an expert in like the full nuanced chemistry. If that reaction is reversible and you can reduce it, then yes, it should be fine. But if it's not reversible, then obviously that site is now blocked and you can't label it.
We still have a couple more questions. I'm going to do just one for now, and then we'll take the rest later and let you keep going. Okay. Okay. But maybe relevant question. Do these labeled quant use a top N DDA?
So they can, yeah, I mean, they can, and there's a lot of flexibility to the experimental design. When looking at an iTrack or TMT labeled sample, you can define the top N, meaning it's going to do MS1, look at all the peaks that are observed, and then pick the top N number, whether it's 10 or 20 or whatever, and isolate those to do MS2 on them. And that will give you the peptide sequence plus the quantitative aspect. What top N you pick depends on your chromatogram, your peak width, and being able to sample as many of the chromatographic peaks as you can without oversampling, meaning measuring the same one over and over again, or undersampling and skipping ones that are maybe too not intense enough. Does that answer that, hopefully?
I think that answers questions. We've got a couple more questions, but I'll hold them so that we can keep going. And I see some proteomicists, mass spectrometrists in the chat that are jumping in to answer some questions. That's fantastic. Please keep doing that. Drop some references, drop your experiences. We're just two people with our own backgrounds, but we know that there's a lot of you out there that are also doing mass spec all the time. Definitely keep talking amongst yourselves, but we're going to continue with the lecture portion.
Thank you. Okay, so the advantages, I think we're on to label-free. So label-free proteomics is often a great place to start because it allows you to see gross changes, large changes between samples And if those are the changes you need to see and make sense from a biological standpoint, that should be good enough. But often you're limited because you might want to be looking at hundreds of samples, and that would take a long time from the label-free strategy. So here the idea is often called differential proteomics. No labeling is used. Samples are run at different times. So while you may be able to batch process them, like in a 96-well plate where all of the digestion happens practically simultaneously, you're analyzing them by LC-MS at different times, one run after the other. The abundance of the peptides you observe are derived from usually the MS1 signal, because you are not frequently and repeatedly fragmenting the same peptides over and over, unless you're doing data independent acquisition, which Lindsay is going to cover in the second half of today. Ideally, necessarily, multiple replicates should be run of each sample in random order so that you are 100% sure or very high percentage sure. The changes that you see are related to biology and not a poor injection or bad electrospray. So you want to make sure the technical variability is assessed by doing replicates. And then you want the quantitation is assessed from a number of varieties of options. Spectral counting is one, spectral intensity, extracting the MS1 or the MS-MS signal if you're doing DIA. And that basically is how that works. So an example here from a label-free experiment, we ran the same normal sample in red 4 times and the same tumor sample in blue 4 times. And because of the nature of LC-MS, particularly using nanoflow, often you get retention time shifts. So that doesn't show the alignment, but often the data can be aligned by looking at retention time and mass to charge features so that they can overlap. Once they're aligned, then you can begin to pull out individual peptide data from the MS spectra. So here's an example doing quantitation based on the precursor, where we've got the 4 blue replicates, the 4 red replicates. MS scans were taken at the little diamonds, and MS events were taken with these big triangles. And we're able to get a ratio based on the peak area of the average of the blue, the average of the red. And then from all of these, you might only get good MS2 spectra for 1 or 2 of these individual runs, but if it aligns with the mass to charge and the retention time and the charge, then you may be able to quantify and just assume it's the same peptide. It's not always the case, but this software gave information to allow some of these to be made, some of these assumptions to be made. So which are the best options? Some people have already started asking these questions, like what are the considerations for designing targeted experiments? Can you quantify by MS? What is the best mass analyzer, if you even have a choice? And then how do you evaluate your data? So the goals of moving from whole global quantitative proteomics to targeted proteomics is you need to understand what answers you need to get for the questions that you have. Are you simply looking to detect something and see if it's present or not detected? Or are you trying to quantify discrete changes in samples, in which case your number of figures of merit, like accuracy and precision and linearity and things, you need the information to calculate these. So this is just a laundry list of considerations in developing a targeted assay. And I'm not going to go through it in total detail because I'm sure people will have just better questions that these we'll get to eventually. But the idea here is, why are you targeting a sample? Do you have empirical evidence based on Western blots, based on ELISAs, based on mass spectrometry? Or is it like a hypothesis like, oh, this would make biological sense? Any of those are valid. And it's always good to try to go in with the hypothesis if possible. I mean, it's interesting to think about what changes cancer may cause, but you can kind of dial into the types of proteins and interactions that are going on with cancer. You may not get it right by guessing 100% of the time, but you talk to a good biologist or cancer researcher, and they probably already have their list of favorite proteins. You also want to think about what amount of the protein might be present in the sample. So we know currently we can measure about 100 or so, maybe less attomoles of a peptide in about a microgram of sample when doing nanoflow on conventional LCMS. That's without enrichment, that's without fractionating. So how much of your starting material do you need to get about 100 attomoles of protein? There are a lot of assumptions of the targeted protein. Sorry, there are a lot of assumptions here, and we're glossing over things like sample loss and digestion efficiency, but this is a ballpark starting point. If a protein that you're looking at requires 10 milligrams of protein, then that's going to be really hard to do without significant sample prep and enrichment. How many targets do you want to look at per sample? And then how many transitions per peptide, because this might control what type of instrument you use, a high resolution mass spectrometer or a triple quad. And then cost. Do you have money to spend on labeling reagents or internal standards, or for lengthy LCMS runs, because those often come at a cost too. And then how much time do you have to spend looking at sample acquisition and data analysis time? And then how unique is the peptide or protein? That's something you can figure out ahead of time by doing literature search and blast searches. And then what are the potential for false positives and interferences? So this is something I just tell everybody when I do these kind of things. Never underestimate the literature. You can sit at your computer and figure out a whole bunch of things about what you might be looking at before you actually look at it. And this really gets down to, sometimes things haven't been published. For example, quantifying a certain protein in a certain disease state, not just because they haven't been thought of or tested, but because there were no positive results or it was just impossible. And I've encountered this a few times in my scientific career. And so that's just something to consider. If you're going to do a literature search, you're like, oh my gosh, there's no information, I'll be the first to publish. Maybe you won't, because maybe it's impossible to get the data you need to make a significant impact. Also, you can look in the literature to see right now, like I had mentioned earlier, we know about our detection limit for peptides in a digested sample by LCMS, give or take. And these are all things that are usually plasma proteins. If you're looking at plasma, high abundance, perhaps not that interesting. And in order for you to get down to lower concentration peptides and proteins, you have to do something to the sample. And so this is a really cool figure, I think, that gets into the different levels of things you can do to your sample and the impact it has. So when we do LCMS of peptides for proteomics experiments, we obviously start with a protein sample, which could be cell lysate, tissue, plasma, any other kind of biological fluid. We usually start with 10 to 20 microliters of plasma, or a little over 1 million cells per sample. This can be dialed down, obviously, now that we're getting to more sensitive instruments, but this is just in general a good amount to have when you first start. You can do protein level enrichment by amino affinity. You can do an anti-protein antibody to enrich just your protein. You can pull out particular aspects of the proteins in your sample. You could do cellular fractionation, you could do size exclusion, you could do ion exchange, and separate your total number of proteins into bins, and then fractionate them analyze them one by one to help get to lower amounts of protein per sample. Another fractionation step is at the peptide level. So after you digest, now you can pull out your phosphorylated peptides through IMAC. You can do amino affinity by anti-peptide antibodies if you have that capability, or you can do fractionation, like I mentioned, cation exchange or basic reverse phase. Now, this step here, this nano LC, had been the major workaround most researchers use to get to that level of sensitivity. You can load less sample and detect more if you're running at a lower flow rate into the LCMS. And for the quantitation portion, often triple quads are used, but also using a targeted list on a high resolution accurate mass instrument will work too. And this allows you to see the whole complement of product ions rather than just picking out certain transitions. This sample enrichment process, I'm just walking through some numbers with you, starting with plasma, just so you get an idea of what they do. So plasma is about 50 to 70 milligrams per milliliter of protein content. So if we want to analyze only a microgram, it winds up translating to 15 nanoliters of plasma. Obviously, we can't work with such a small volume, so we start with more and then dilute before the analysis. But if we were to do a protein level enrichment, so for plasma, let's take away the top 14 most abundant proteins to look at the more interesting ones. If we bring that sample after depletion back to that same starting material volume, it's about 220 nanoliters of neat plasma equivalent per injection. If we do the orthogonal peptide level chromatography, now it looks like the equivalent of what we would be loading if we hadn't done the enrichment is between 500 and 1500 nanoliters. So you see, even though we can't load that much on a nano column, the equivalent we're able to see gets us down to proteins that are so low abundant, we normally wouldn't detect them without this level of enrichment. And then antibody enrichment, like anti-peptide antibodies, allow us to just pull out those peptides and ones super similar to them, which is like a tiny little subset of the whole sample. And this can translate to starting with a volume of a milliliter per plasma, and then taking all of that and injecting onto the column. So this is the effect that the different levels of fractionation have, and kind of understanding where you need to go will help drive that. Also, the more separation processes you put your sample through, usually the better signal to noise you have. And this is due to a relationship of, with every mode or stage of analysis or stage of separation, like 2 is LCMS, GCMS, so you're just separating and then analyzing, or MSMS, fragmenting and looking at those data. Unfortunately, there's signal loss associated with increased stages of analysis, but the noise usually goes down quicker. So these are just examples of how you can hyphenate different modes of separation, all amenable to one another, to get to improved signal to noise for your detection, which is ultimately based on this selectivity idea. So just for those that are like, I hate nanoflow, but the reason that we go to it is looking at this sensitivity improvement on the y-axis. So this is looking at flowing at a higher flow, starting with a 2.1 millimeter scale. So you can load a ton of protein onto that. But if you take and dial the flow rate back and adjust the column inner diameter from 2.1 to 1 to 300 microns to 150 to 75, what you see is this gain in sensitivity, this gain in signal to noise. And this allows you to have better quality data and confidence in what you're doing for identifying those proteins and quantifying low abundance ones. So if anyone just ever is like, why don't we do high flow? High flow is great, and you can bang your samples through in a short period of time, but often you're giving up the sensitivity in certain analytes that you wouldn't detect otherwise. So this is just a little history of how people have been quantifying by using MS as a mass analyzer and detector. And this is nothing recent. It started a long time ago. And you can see over the decades, these are the 2002 was Cylac, 1999 was the ICAT, iTrack came about from there. But inherently, I would say, as being on Team Mass Spec, mass spectrometry is not inherently quantitative. There are a lot of things that affect it, and there are some unknowns in our calculations for being able to quantify things. So if we compare this to Beer's law, which is for spectroscopy, we get an absorbance from a signal, and we have a known extinction coefficient we can usually calculate based on the properties of the molecule we're looking at, and a concentration which can be measured based on that signal and predetermined extinction coefficient, and then a known path length. So all of these things at any given point can be set up so that there's only one variable, and we can measure the rest, or the rest are known, and calculate that one variable. Here on the MS side, we've got analogous, some signal, but the concentration is affected and has this ionization coefficient or transmission coefficient or something that affects not just maybe the concentration of the analyte, but the concentration of the sample that it's in. How many other ions are being produced at the same time, and what is their effect? And this K value is largely unknown unless you go through and try to empirically determine it. So it's hard for us to do something like change the concentration because we don't know if this ionization coefficient is changing at the same time or not. And making assumptions often makes you just have wrongly interpreted data. And so this is where using the labels helps us to sort of overcome this ionization issue, particularly if we use labels that are for the same peptide, but label them a little bit differently. So this is one example where we use stable isotope dilution, and we have these amino acids in the peptides we're looking at. Trypsin clips at lysine and arginine on the C terminus, and so the majority of the peptides in our samples digested with trypsin have lysine or arginine on the C terminus. We can take and label the carbons and the nitrogens in these amino acids and synthesize peptides for the peptides we want to target, and then spike them in known quantities. And so this kind of came together in the mid 2000s and made it really cool because it just worked. Then when we take the peptides and we fragment them, we tend to fragment across the amide bond next to the carbonyl and this nitrogen, and we generate B ions where the charge is retained on the N terminus, and Y ions where the charges are retained on the C terminus. And these numbers allow us to assign fragments of these peptides starting from the N terminus or the C terminus and add up the amino acids to look at the whole mass and see a loss of an amino acid, and then you know what amino acid was lost based on its mass. So this allows us to go through the sequencing of each peptide. And this is just an example of a light and a heavy peptide where the phenylalanine, in this case, this is super old. I was in grad school last decade. Anyway, this phenylalanine was the heavy labeled version. And so the fragments here shown in red, the light is 1031 and here 1037. We had 6 carbon 13s on that phenylalanine, and that's incorporated into the B ions. The Y ions here don't have a change in mass because this would be just the proline and lysine, which are not isotopically labeled. Now we usually label these C terminus, so all of the Y ions that we see have that signature mass shift, and we can quantify it on all of the Y ions. Okay. This is just kind of reviewing before looking at stable isotope dilution. You spike in your standard in a known amount and then quantify the peak area relative to the light peptide, which is coming from your sample. And this is how we often use Skyline to do this. And the idea here also is quantitatively, you should see a linear response for changes. So we look at often the ratio of the MS response of the analyte divided by the internal standard, and the ratio of the amount of these that if you spiked in a fixed amount of internal standard and a varying amount of light, then you should also see when we do the light versus the internal standard, this slope with a regression line of y equals mx plus b or y equals x, because usually in this case, the slope should be ideally 1 and the b, the background signal, should ideally be 0, but that's often not the case. So we like to generate these lines and look at the slope and the y-intercept to see if they have any significant impact. Here, one question people may have is like, oh, I don't want to analyze, I don't want to generate peptides with like a individual internal standards for all of my peptides because it's expensive. And so if we take a step back and look at the impact of what having an internal standard versus label free, and then an intermediate, which is called a labeled reference peptide option. So this was a response curve that was generated all like individual concentration points, obviously, and these are plotted as measured concentration in femtomole per microliter of that peptide to theoretical concentration, what we think we actually spiked in. And here on the left-hand side, we had an internal standard. So the measured concentration is that analyte divided by the internal standard. multiplied by the amount of heavy that was spiked in, and this gives us the femtomole per microliter concentration. Then when it's plotted, we look at the R squared and the slope, and that's all fine. But the coefficient of variation, which means the variability of each of the measurements taken, is about 1.2%. So we're able to get very reproducible measurements when we normalize a peptide by its internal standard, same sequence, just internal standard. If instead we take these same data that were generated here for the light curve, but instead of dividing by the isotopically labeled peptide that we're looking at, we use a different labeled reference peptide that appears somewhere else in the chromatogram. So it's a different sequence, a different retention time, different ionization potentials, different fragmentation. But we can use the same one peptide internal labeled reference for all of our peptides. What we see here is when we plot that slope, our coefficient of variation on these data points is now 5% instead of 1.2. And so we also see there's variability down at the low end and at the high end that this labeled reference peptide can't account for because it's occurring somewhere else in the analysis. And then last, label free. So let's just say we're targeting a bunch of peptides and we don't have an internal standard. We're not doing any kind of normalization. This is the same exact data that was in A and B. So we're looking at the same peak areas of the light peptide, but now they're not normalized. And now we see for these replicates, there are data points that are dropping down, and perhaps we don't know why. Perhaps there was like one replicate was just not so good. And because we can't normalize and take that variability away, now the coefficient of variation is up to 13% or 0.13. And so understanding that this is all the same light data, the impact that having an internal standard has is accounting for that variability and improving the accuracy. Okay, so this goes through some questions of like, what do you want to target? Do you want to go through and have empirical LC-MS data first? Do you start with pure protein standards? Do you synthesize peptides? Are you getting your protein targets from the literature? And then how is that literature data produced? Do you have access to it? Can you look at it and see how this will influence your experimental design? So it's always good if there is data to consult it, particularly if you can't generate your own right away. And peptides, they fragment similarly, even on the different mass analyzers. So, and this is just based on if you're using the same type of fragmentation mechanism, meaning collision induced or collisionally activated dissociation, you should see a lot of the same fragments, perhaps not at the same ratios, but you can be pretty confident that they will be generated similarly. So now, if you have empirical data, what do? We search the data against proteomic libraries. You can identify peptides with certain confidence. You can generate a list of these peptides, like the ones you're most likely to detect, and then target them. Whoops. There are some resources you can use to sort of seek out this information. There's a lot of publicly available databases. Panorama is a great one for looking at targeted workflows. Passport was generated a while ago, and this looks at the most intensely observed peptides after a tryptic digest of a particular protein. So this is based on empirical information. Point being is you would want to target peptides on the left-hand side of this plot rather than on the right-hand side of this plot, particularly if you are not seeking out post-translational modifications or certain site-specific modifications. And then there are peptide and protein targets from public resources like the CPTAC assay portal. And this goes down into the peptide sequence from a particular protein, limit of detection and quantitation, precision and repeatability that have been achieved empirically. If you have no idea where to start, you can do in silico digestion and you can generate the data and you can generate in silico versions of the targets that you would be looking at. So you start with the general rules, and these are some settings in Skyline that you can set up for doing digestion and looking at numbers of miscleavages, what the optimal peptide length is, make sure it's not close to the end terminus of the protein because you might have exoproteases that are going in and chomping up your protein. Generally, we like to exclude labile amino acids like methionine because it's prone to benchtop oxidation, which means now your peptide is split into the non-oxidized methionine and the oxidized methionine, which is different by mass to charge and often retention time. So point being to beware of post-translational modifications. You can usually look up and see if your protein may have glycosylation or phosphorylation or something based on a consensus sequence or something in there where it's been determined to exist. So here's where we can go into some examples of how to use Skyline, data dependent, running a database search and identifying peptides and then generating that spectral library in Skyline. So this is kind of what we did with the plasma data set that you should be able to download. From here, we can select a certain number of targets based on what we want to track in a number of samples and go through and sort of refine this process. So for people using triple quads or SRM, we often want to take the total number of fragment ions and narrow it down to like 3 or 5 most intense that may not be interfered with. And this requires a little method development, but once those are set, they're pretty good. For PRM or parallel reaction monitoring, you can keep all of those and you can just worry about how the data looks and then not include some of those peptides at the end, but they will always be collected by the mass spectrometer. And ultimately, by going through this process, you have a refined transition list for method optimization. So to guide you through some of the, I guess, hiccups or things where you're like, oh, this is my first time doing this, I don't know what to do. So this just walks through some examples in Skyline of data that we generated, and we're looking at the MSMS spectrum. So when I set up a list and I'm looking at like, what's the best peptide to pick from proteins, I will usually look at the +2 charge state and the +3 charge state of a particular peptide. So here on the left-hand side in what we call the tree view, we've got the protein accession number, the peptide sequence with it, which is EHVAV, ... And then the +2 charge state at 617 and the +3 at 412. And then we've got a bunch of product ions for each one. These were all automatically generated in Skyline by giving it a certain number of criteria, like give me 4 or 5 transitions, give me the 2 charge states. And so that's what we see. Then when the spectral libraries are brought in, we can observe them this way, visualize them plotted. You can have Skyline label all the peaks that are observed. So these were already identified by a spectral database search. And then that result from that spectral database search was brought into Skyline. And now we're able to see these peptides. So we see unreacted precursor in the 2+ charge. We see a whole bunch of product ions. It looks pretty good. But we also see information for the +3. You're like, oh, well, at a quick look, you're like, well, the +3 isn't generating so many product ions. So maybe I don't want to pick that. But if you look at the scale on the +2 and the +3, the +3 is 1 million and more signal, whereas we are at 1 million and less for the +2, even though it generates more product ions. And so this is really one of those things where I don't know what to do. Initially, I might keep both of these, and then when I wind up getting it into a actual targeted workflow, get rid of one of the charge states. But for right now, we often like to try to pick higher mass to charge fragment ions because you can see up here, there's not a lot of chemical noise that's coming through with these signals. Whereas if we had to pick the charge 3, we would be picking ions down here where there are a bunch of little gray unlabeled peaks that are going to be generated that have nothing to do with our peptide and may increase the noise of our sample signal, thus decreasing the signal to noise ratio. But again, I often keep a bunch of them in until my last phase where I might delete one of these charge states. So also some peptides are super beautiful. So when we go through, we're like, this is a lot of ions that are generated. This looks great. I'm pretty confident that this is the peptide. But some peptides don't look great. And so even though this was identified based on database search, and it got whatever score based on my cutoff criteria, it's not generating ions that I think are worth pursuing. If there were other peptides from this protein, I might just delete this one and not pursue it. But the point being is not all the data you get are perfect, and you might be left scratching your head with like, why was this identified? It's not, doesn't look that great. But for the best results, you want to explore the data to understand it. Use predictions, algorithms, literature as your guide, and then also check the data manually. You might see great, you know, this peptide might have come through as like, oh, this is a great hit, but you wouldn't know really what it looked like until you looked at the MS2 scans, which are not often presented in a proteomics experiment after you do the database search. And then once you do this, like every step, every experiment you do, you'll become experienced in generating these targeted methods and become more confident in your data. And just a quick little analogy, there's this, you know, little story about different people touching an elephant, touching a different part and thinking it's something different, like this when they're all blindfolded. So this one person's looking at the leg saying it's a tree, one's touching the trunk saying it's a snake, one's saying it's a spear. And it really just depends on your perspective and how much information you're able to take in. So if you're able to look at everything, you might be able to identify it as one kind of elephant or another kind of elephant. Mr. Snuffleup, I guess, has no ears, so that would be incorrect, even though you might have identified it as an elephant because it has a trunk. Or, you know, sometimes the data are clear enough where you're just not identifying something. So let's see, I'm going to pause there and see if we have questions before going into the next phase.
We do have a ton of questions. I guess, how are you feeling for time? I think we've got about 20 more minutes. Is that right? Let me check.
Yeah, so here, what I was thinking is we've got the hands-on, and we can do the hands-on, and we can go through kind of individually. Or if people are not wanting to do that, we can go through just additional data and I can walk you through how to do some things without doing it live. So it's more of a, what do people feel?
How are people feeling? Maybe a couple, how are people feeling? Drop some feelings in the chat. Continue lecture or go to hands-on? Do it live. Hands-on, hands-on, hands-on. Continue, okay. Live, hands-on, hands-on. Getting a lot of votes for hands-on.
Seems like we've got some hands-on. So has everybody opened up, downloaded their plasma proteomics thing from the websites that I posted? Tutorial.
We did have a couple people say that they were having problems downloading. I don't think it's on our end. I think it might be some antiviral. Maybe some people are running like an antivirus for their computers, and maybe that's blocking the download, because I think other people have had no problems.
Okay. Hold on, I've got to extract. So I hold on a second. Let me share just so y'all can go through this with me. So I just downloaded this from the website and I got a zip file, and I'm just going to go extract all. I'm going to pick something. I'm going to pick my documents folder and we'll just put it in there. Extract. Okay, so in my documents folder, I've got a plasma directory. So I haven't renamed or done anything. Hopefully y'all can get along this far. And then there's also this plasma tutorial, which is the PDF that walks you through everything. That's not it. Let me get rid of that. No one needs to see that. So I'm just opening up the plasma tutorial now. I am sharing, right? Can everybody see?
Yep, we can see the PDF.
Okay. So here, just to walk through some, well, let's go back. The idea here is we've got 6 proteins that we're going to be looking at, and all of the materials you need are in this plasma directory for this workshop. Lindsay, you made this tutorial up, right?
Correct.
Thank you. Everybody thank Lindsay. So if we go, yeah, I think we'll first, we assume we know nothing about these 6 proteins. And that'll be the first way we're going to start off, because I'm assuming nobody knows anything about these proteins. So you can build an assay without any prior knowledge, because you're going in and you're looking at information about the IDs, you've got your identifiers. So I think, if I'm right, we have, this is how you would get the FASTA files for your proteins and load the sequences into Skyline, because Skyline needs that information in order to generate peptides from the protein sequences, or you give it a list of already clipped peptides from the digestion process. All right, Sophia, you have raised your hand. If you want to come off mute and tell me what's going on, we could do that. Nope, the hand's down. Okay, maybe it was an accident. Everybody so far kind of going here. I would say, if we go to this plasma folder, there's a plasma standards Skyline setup that should already, you have to extract this. Make sure you extract this before you just go into a zipped directory to open it up. But you should be able to go in and open the Skyline document by double clicking. All right, going to remind me later. Then we've got this Skyline file. Now this has been pre-populated for all of you with the protein accession number and the protein sort of code name, and then peptides that have been filtered based on digestion settings. So what I'm going to show you first is, if you hover over this, if you hover with your mouse, you click it and you hover. Nope, you click it. Oh yeah, see, so this window pops up that has all the information that Skyline has pulled from the FASTA file. So the FASTA file has the protein name, accession number, preferred name, the gene code, the species it came from, and then the long text description. And then it has the sequence of the protein. So one thing I want to point out first is this one has an N-terminal methionine, which is usually the signal sequence, and often proteins are processed after translation and have this methionine removed. If you're not sure of what state this protein is in, I would say don't target any peptide that includes that first amino acid in the N-terminus. And that's why when we go through the in silico settings of like peptide settings, so settings, peptide settings, digestion, you can include different proteases. You can include maxed miscleavages. I usually have 1 or 2. In the prediction tab, nope, I don't want to do that yet. In the filter, this is looking at the minimum and maximum peptide lengths, and then exclude the N-terminal amino acids. So here's where I would put in like at least a few amino acids to exclude at the N-terminus. Okay, everybody good so far? All right. And then for those of you that haven't seen Skyline previously, if you click on that protein name like I have, what that's showing me, excuse me, is it's highlighting all of the peptides from that ApoA1 that have been detected in the chromatogram. So if I left click and zoom in, I can get a bit of a better view of these peptides, and it's labeling them based on the first 3 amino acids or however many is needed to make it a unique peptide ID. And we can see these are all nicely separated. That looks great. If we click on the first peptide that has the DLA sequence, what's shown here is a couple of peaks in the chromatogram where something has been picked at 9.18 minutes, but there are some other peaks here. So something I like to do is like, let's zoom in, again, left click, or if you right click, you can do auto zoom X axis and make it best peak. And that'll zoom into the peak that Skyline thinks is your peptide. So what's nice about this is we see the retention time, we see the signals from all of the product ions. If we right click, we can put in, hold on, where's properties? Legends. We put the legend in, we can see the product ion signals that are being extracted from the data and their different colors. And then if we click on each of the individual product ions, it'll highlight them. And so normally if we were doing this live and we had the raw data and we had to import it, you would see how many points across the chromatographic peak are being counted. That's what this little red question mark is. Unfortunately, sometimes when you zip and transfer Skyline files with the raw files, that connection, depending on how you unzip it and load it up, can be lost. So this one isn't registering right now, but you could go through each of the peptides and see how they look. Also, these peak area indicators, these little green balls, they're showing you that there is signal for each of these product ions. And more than 50% of them have been detected. So that's a green signal for the precursor. Even though it's not actually measuring the precursor, all of these signals are kind of give us a confidence of what's being detected. And so that peptide too. If we get to a point where there's no peak area indicators, like let's go to ApoA2. There's no data here. That's because we're not actually collecting data in that one file. So now I'm going to move to the ApoA2 tab. So this is the ApoA2 data file. And now we're collecting peptides from ApoA2 in this run, but not ApoA1. Let's see. What else can we do? If we click on edit view, modifications, protein, we don't want that. So let's go through some of this other information right now. So also, those of you that are new to Skyline, I have these data should probably be pretty standard when everybody opens it up, set up. I have the chromatograms from my individual protein runs on the lower portion. On the upper portion, I have the retention time plot. So there are a lot of different retention time things you can look at. If you go to view retention times, I can look at the peptide comparison. And so this is adding a tab to that area, and it's looking at all the different retention times on the y-axis and all the different peptides on the x-axis. Then I can go through and sort. Right now it's sorted based on that peptide's location in the document on the left-hand side. So if I sort it based on retention time, now I see that my earliest eluting peptide is this first one, and I should be able to click on it. And it'll highlight it in the left-hand side. And it's this peptide SQAIA. It's in a different protein. We can kind of go figure out which protein it's in by clicking. There we go. It's in the ApoB. All right, and that's the nice peak area. Oh, it's also shown right here. Okay, we can also sort these based on, whoops, peak area. That's more random because now we're looking at the retention time based on peak area, and we would expect the peptides to just be all up and down based on their peak area. In the peak area plot, if we go to view, there are also a lot of different ways it can be plotted where for the replicate comparison, this is the view that we're observing right now. It's most ideally used when you are targeting the same peptide over a number of runs and you want to see how it changes over a number of runs. In this particular Skyline file, every run is for a different protein. So this view is not so useful for us in this setup. So if we look at peptide comparison, now we can look at all the peptides. Let's see. For the different runs, and how they compare based on peak area. So I don't know if we have to like zoom in here. Yeah, so if we zoom in, or we could put this on a log scale, you can see all of the log peak areas on the y-axis and see how their intensities change with each of the peptides. And so if you click on a peptide, it'll bring you to that peptide, and we just need to go to the proper protein to see the signal. There it is. All right. Does anyone have questions on things they want to get from this data set but haven't figured out yet, or similar things they want to get from their own data sets that we can use this for, and we can kind of get a couple specific questions hammered out?
Got lots of questions in the chat, but only one of them is maybe regarding this data set. There's a question asking, what exactly is a scheduling window and what do you use it for?
Oh, okay, that one I can show. So a scheduling window is shown here. And if you think about when you target a list of peptides and you have 5, it's pretty easy for a mass spectrometer to, one right after the other, target each of those 5 in like a cycle. And like 1, 2, 3, 4, 5, go back to the top, 1, 2, 3, 4, 5, and you'll get enough chromatographic points across your peak. Even if the peptide's not there, it's not spending a whole lot of time on each individual peptide. And so not a big deal when you have a low number of peptides to target. Once you have a high number of peptides to target, like 100 or 1,000, that means your instrument's going to spend so much time in a given cycle on peptides that are not actively eluting, and you might only get a couple of points across your peak, because let's just say, for argument's sake, it takes like 10 milliseconds per each of these transitions. The more you have, the more time it's going to spend in a given cycle. So what you do is you schedule your peptides. And I think I actually have, I think I have a plot that kind of gets to this here, because this is one of the things. So this is looking at, can everybody see? Oh, here we go. So why do you want to schedule? We can't always detect all of the peptides in a given cycle, so we need to split them up to when they're eluting, and spend more time on peptides when they are eluting. And we get the elution information from discovery and from setting up method development and optimization runs. So let's just say we've got this one peptide at 28.3 minutes, and we're only going to collect it as it elutes, and we're going to put a little window around it. Now, retention times shift, like what this is showing, as we do our runs, the peptide's moving earlier and earlier, and we start having it go outside the retention time window where we've programmed the MS to look for it. And so we want to set these windows as wide as we can without spending too much time waiting for a peak. And so these plots are what give us that idea of, if we put a 10-minute retention time window around each of the transitions, and we have 325 transitions, this is going to spend a whole lot of time generating data for transitions that are not actively eluting. And so we're kind of looking at the scheduled time here and the number of concurrent transitions. Each of these transitions, if I said it takes 10 milliseconds to acquire, then 10 milliseconds times 100 transitions is 1 second. So that means that this 2-minute retention time window, we're at about a 1 second cycle time to target 80 transitions here. And if we go longer, then that means we're going to get fewer data points across our chromatographic peak with that same dwell time. And so what we try to do is set these parameters up so we can see how many concurrent transitions there are, or precursors if you're doing PRM, and making sure that we're spending the right amount of time on these as they're eluting. So if we look at the Skyline file here, this is looking at concurrent transitions, and we could set a 2 or 4 minute, or even a 5-minute retention time window around these peptides for this run, and we would still be able to get enough points across our chromatographic peak. Okay. If I can go through the questions, how does utilizing multiplexing influence detection of low-level peptides? So if you use multiplexing and that peptide is not present in all of your samples that you've combined, or it's very low abundance in some of your samples, but high in perhaps the carrier channel, you will be able to still identify it. And then the quantification from the reporter ion will allow you to get a sense of where it stands relative to all of the other samples that you're multiplexing with. If you did label free, it may not even have been triggered for MSMS, so you would never have identified it. So that's one of the main benefits there. See if doing targeted MRM assays on a UPLC triple quad, what adaptation to the instrument yields the most significant increase in sensitivity? Yeah, you can do microflow on modern UHPLCs for sure, or even higher flow. And it really just depends on if you can detect the thing that you're looking for. And the important things here are chromatographic resolution, narrowing the peak. So columns that use super small beads will always have a good resolution effect based on, like less than 2 micron particle beads versus columns that use 3 micron. Their peaks will be broader under the same chromatographic conditions. The most significant increase in sensitivity is often lowering the flow rate and moving to very small chromatographic diameter beads or longer columns that will improve your chromatographic resolution, or using orthogonal approaches like ion mobility or FAMES usually increases the signal to noise for MRM. SRM and PRM. So SRM is selected reaction monitoring on a triple quad, which means you're individually looking at the Q1 setting, so the precursor, and one product ion at a given time. And the instrument will always look at things in pairs, Q1 and the second quad, or sorry, Q1, Q3, and kind of go through the list. So in a case like what I'm showing in the Skyline setting, the 626, it looks at all of these product ions individually, and that's how a triple quad runs. Whereas with parallel reaction monitoring on an instrument like a TOF or an Orbitrap, it's generating the product ions all at the same time. And so in one scan, you get all of them. And so that's called parallel reaction monitoring. I see we're a minute over, and Lindsay is up next. I will be hanging out online to help continue answering questions. So I'm happy to do that, but I don't want to take away the Lindsay time for the next session.
Yeah, we've got a 15-minute break now. So feel free to get up, grab your coffee, grab a beer, depending on what time zone you're in. I won't judge. A little Irish cream in your coffee. We're all friends here. We'll stick around to answer questions. Reminder, try to make sure to use the Q&A in the chat. And we will get started again in about 15 minutes.
I'm happy to hang out during this 15 minutes. If anyone has questions, I can answer live.
Yeah, we've got lots of questions in the Q&A. Brendan and I have been cleaning up maybe some of the easier ones.
Okay.
But if you have any of them that you want to address.
All right, Brendan's on that.
And recaffeinated. We started at 8 a.m. over here on the West Coast. Oh, I think to unmute, I think I'm a host, so I can do that. Do you want to be my guinea pig, Lee Foster? Awesome. See, I just sent a request.
I accepted. Can you hear me?
Yes, we can. I did it.
Great. Hi, Lindsay. Yeah,
How's it going?
It's going all right. I'm home today with my little baby. He's kind of sick, but still here and still here and really interested in what you guys are doing today. So maybe since we're taking a break, I can ask a couple of questions.
Yeah, of course.
So I am moderately familiar with Skyline, but I've always been kind of confused by the verbiage. So maybe you could just help me understand when you say transition, what is a transition?
You want me to do this?
You can do it. I learned it from you. It only seems appropriate.
Let me see if I might have a slide that. So a transition really is just talking about when you fragment your peptides, you start with a precursor and you wind up with a piece of the precursor or product ion. They used to be called parent-daughter. That is not the most up-to-date use of the terms. Now they're called precursor product, or transitions is the pair of those. On a triple quad, you analyze those in the first quad and the last quad. So usually Q1 and Q3, and that pair is called a transition. In PRM, it's pretty much the same thing, except they're not really quads. It's just the precursor and the product ion pair setting. So it's the transition from the whole to the piece.
Okay, understood. Thank you.
Mm-hmm.
I did have one more question, particularly on the data set that we were just looking at. So first of all, the data was beautiful. I don't know if you guys were using like purified proteins or what, but one thing that I noticed is that for some of those peaks, one of the Y ions was significantly more abundant than any of the other. And it's not something that I've regularly run into when I've been looking at data. So I'm wondering if you could speak to one of those ions being so much more prevalent and what that means for the data set.
Hang on. I might have examples of like this. So basically, what I see when I'm looking at data sets and I'm trying to figure out what signal is the one coming from that particular peptide. In the case of Skyline, it's pulling out all the product ions, right? Usually, when we digest with trypsin, we usually get a lysine or proline at the end, and there's often, lysine or arginine at the end, so a K or a R. Mm-hmm. If there's a proline before that K or R, then the intact peptide usually like fragments there just due to the nature of the proline, and that becomes one of the product ions that's generated with the least amount of energy, or it's like most quickly to fall apart versus all the others. And so the Y2 product ion, if it is that KP or KR, or sorry, PK or PR, you'll see this big signal because it's just the easiest to fall out. You shake it, it falls off, right? Okay. So, and that's also one of the reasons we don't normally like to use these small Y or small B ions, because they come from 2 amino acids. And if it's the Ys, we know that 50%-ish of the peptides have a lysine and 50% have an arginine. And so it's easy to miss a sign based on that. Usually the way I like to look at it, I'll just share my screen real quick and look at the Skyline file, is when we are looking. So for people who are wondering what Lee's talking about. Yeah, so this is a Y9, which is the most intense. I like to try to see that the other product ions are at least 10% of the biggest one. And sometimes you just don't get lucky with that. So in this case, I don't know why the Y9, it's something in here is fragmenting to generate that. But it is one of those things where you get what you get. and you try to see if it makes sense with the sequence of the peptide. But it's not always an ideal, nice distribution of all intense product ions.
Yep, understood. So in the scenario where you're talking about like a proline next to a lysine or an arginine, would that be addressed if you were to set the exclusionary criteria that you don't have any miscleavages?
It could, depending on if it generates a miscleavage or not. So usually the KP or RP would be the sequence where prolines after the lysine cleavage point, that would be a miscleavage product, not if it's before the lysine or arginine.
Right. Okay, and just like my knee-jerk reaction when I see one that's very far above the others would be to exclude that particular ion, even though it's very high intensity. Is that something that you would suggest, or is there a particular benefit to having that particular ion?
So in my opinion, I would base it on what your needs are. If that is the only signal in the entire chromatogram you can see, there's no other peak that would be like a second best, then I might keep it. And if it's particularly the only peptide for that protein, I might keep it until I'm done with the method development and make my decision to keep it or axe it at the end. Because you might have some peptides that are looking inherently more stable, but, more equally distributed, but they might not be stable after doing run to run and having them sit in your autosampler vial for a while. Whereas one that has that weird distribution of like one super intense and the rest not, that may be like your best peptide you get. So I don't usually axe things early unless they look horrible and there are no other backup options.
Okay, thank you. I know Lindsay is going to get to more of this, but thank you for answering my questions in the interim.
Yeah, I'll definitely make sure to build in a bunch of time for some Q&A at the end so that we can go back and dive a little deeper into any of these sections that people thought were particularly interesting or relevant to their work. And I think everyone should be back from the break at this point. See if I can get set up correctly. It's always a little bit of a gamble. If I can get my chat and my Q&A up at the same time, we'll see if I can multitask it. All right. So welcome back from the short break. If you left us, if you stayed with us this whole time, we had a little bit of discussion during the middle, tried to get to some of your open questions from the first half. I'll try to also do like a little bit of a review from the first half, a little bit more like kind of higher level, like we gave you a ton of information. The point of that is not that you should know every piece of that inside and out from, like from the heart, you know all of that memorized. It's more to be a reference for you so that as you go either into proteomics or more in depth into proteomics, these aren't new things that you have to Google. You'll be like, oh yeah, I vaguely remember someone mentioning that and it was a quantitative technique. That's kind of the idea. So in this section, I'm going to go a little bit more into data independent acquisition in the lecture portion. And this is to help set you up for tomorrow and Wednesday when Brendan McLean is going to be walking you through doing more in-depth Skyline for data independent acquisition. And then in the hands-on section, I dropped the tutorial download in the chat. The hands-on section, we're going to do a little bit more of a deep dive into looking at data and curating data in Skyline. So I will try my best to keep up with all of the various chat and Q&A, but Sue, Brendan, anyone, feel free to interrupt me if there's a lot of questions in the chat. So as I mentioned, we're going to talk today, this section, a little bit more about data independent acquisition. I'm going to give you a lot of information, but there's really kind of 4 key things that I hope that you take away from this lecture. The first is to just know some of the key differences between data independent acquisition, some of these other acquisition methods that we mentioned this morning. Second, I want to give you a little bit of exposure. There's a lot of different methods out there, but I just want to expose you to some of the various kind of flavors of DIA that you might come across. Then we're going to talk, we're going to spend all this time talking about how great DIA is, and I use it for everything. I'm going to tell you why there's caveats for DIA, and you might not want to use DIA for everything, either because of the way DIA is inherently set up or the way the data analysis is done. And then hopefully by the end of this, you just have the ability to kind of evaluate when there might be benefits for using data independent acquisition and when there might be limitations that make using, your classic TMT or your PRM or your SRM targeted analyses more appropriate. So this morning, to kind of summarize a lot of the techniques that Sue discussed, when we're choosing how to operate the mass spectrometer for proteomics, we're choosing three kind of things, and we're going to compromise something. The first is how scalable the throughput is. So that's how many samples you have to run. If you have to run 6 samples, or do you have to run 600 samples? That's going to affect the way you want to run your mass spectrometer. These slides will be made available right after this. I kind of, as we went this morning, I beefed up some sections that it seemed people had more questions about, and I took out other sections to make room for that. So it was a very dynamic process, but I'll get the slides up as soon as we're done with this. So scalable throughput, first thing we have to think about, how many samples are we running? Second thing we have to think about, how comprehensive do we need to measure the proteome? So this is how many peptides, how many proteins do we have to measure? Some research questions, you really only care about one protein or a handful of proteins. Other research questions, you want to know every single protein that's in the sample. And then finally, how sensitive of quantification do you need for your experimental purposes? Sometimes you might not really care. You just kind of want a general idea. Is this peptide more abundant or less abundant? Some questions are very clinical, and you have to be able to say with high certainty that there is 5 picograms per deciliter of blood of this biomarker. So these 3 things are going to dictate which way you choose to run your mass spectrometer. You'll also be limited by what hardware you have, of course. If you have lots of samples and you need very sensitive quantification, but you don't need to measure a lot of proteins, you're going to choose something like targeted mass spectrometry. So this is things like that, SRM, selected reaction monitoring, PRM, parallel reaction monitoring, or MRM, multiple reaction monitoring. So SRM and MRM, those are performed on a triple quadrupole, versus a PRM is performed on an ion trap, on an Orbitrap, that kind of instrument. So this is, you have a very limited number of peptides or proteins you're interested in. You want to measure lots of samples, or you want to measure very high sensitivity quantification. On the other hand, if you don't really care that much about the quantification, or you care more about getting kind of a survey, a list of all the peptides and proteins that are present or absent in your sample, you might do what's called discovery mass spectrometry or shotgun mass spectrometry. This gives you that list of how many proteins and what they are in your sample. You can do a lot of samples this way, and you'll get that protein list, but the quantification is not going to be particularly good. This is kind of those DDA-based methods. So finally, Sue mentioned these isotopic labeling approaches. So these kind of combine the ability to get that protein list with better quantification than if you just did a regular DDA experiment. So she mentioned things like TMT and iTrack. These are kits that you purchase, you prep your samples, you label your samples, you mix them together. Typically, you then fractionate. So even though you mixed them together, now you have the exact same number of samples that you started with. You measure them by DDA and analyze them with your software of choice. Lots of different software options for DDA. The downside of doing this isotopic labeling is typically you're kind of capped at like 16 samples. So if you need to analyze 600 samples, for example, this might not be your best choice. So we're going to talk today about data independent acquisition. A lot of people in the mass spec proteomics field, including myself, feel like DIA is a good compromise between all of these. You can measure hundreds of samples you'll get the full list of all the proteins that are in that sample, and you get very sensitive quantification compared to some of these other methods. So we're going to visualize DIA using these kind of ion maps. So in these ion maps, going up and down vertically, it's the precursor MZ. So just imagine a very low MZ value, mass to charge value, and then it increases all the way up to very high MZ values. So very, you can kind of think about it as very small peptides at the bottom and then very large peptides at the top. And then along the horizontal axis, going left to right, is the retention time, that LC gradient that Sue had mentioned. So at the very beginning on the left-hand side, those are very hydrophilic peptides. And then on the right side, there's very hydrophobic peptides. So the first thing you might notice in this ion map is this gray, that's the intensity of the peptide at that particular coordinate in retention time and precursor MZ. And you see there's a little bit of a trend, right? The density of the gray in the ion map kind of tends to increase in precursor MZ as you increase the retention time. Large peptides, which tend to have high MZ, tend to also be more hydrophobic than small peptides, which have low MZ. That's the trend that you're kind of seeing in this ion map. Now, if we were to do a parallel reaction monitoring experiment, we would pick predetermined MZ values. So that's on this y-axis, that's picking very specific MZ values that correspond to peptides that we know we want to measure. And then with and then in retention time, someone's walking their dog. It's very interesting. And then for retention time going across the x-axis, we might do scheduling. So Sue had mentioned scheduling as an approach, and this just means if we were unscheduled, then for that MZ value, we would monitor that MZ value. We program our mass spectrometer to look for that MZ value for the duration of our liquid chromatography gradient. Say we're running a 60-minute gradient, we would tell the mass spectrometer, monitor MZ value 1, 2, 3, 4, over and over and over again across the whole gradient. However, maybe that doesn't make sense because we know our peptide should only elute at one spot. So we can tell the mass spectrometer, don't bother looking for this peptide at the end of the gradient because we know it's going to elute at the start of the gradient. That's all scheduling means. So it's just a way to squeeze a little bit of extra performance out of your instrument. So parallel reaction monitoring, you see in this ion map, we're not covering a lot of the space, a lot of the proteome space, because we don't care about most of the proteome space. We only care about very specific parts of the proteome. Sounds like a lot of people are also very familiar with data-dependent acquisition. So if we look at the same ion map and we look at a data-dependent acquisition, at every kind of chunk of retention time, we're only going to trigger precursor MZ at spots that are the most intense. So we tend to cover more of kind of that center ion cloud and don't tend to cover more of these outliers because we're only picking the top 10 most intense parts at any given retention time across this gradient. So finally, with a data independent acquisition, you see it kind of almost looks a little bit more like a parallel reaction monitoring, but tilted, just 90 degrees. So with a data independent acquisition, instead of scheduling the instrument for specific chunks of retention time, instead now we schedule the instrument for chunks of MZ, and then over retention time, just keep monitoring across that cycle of MZ. So it's more systematic, it looks more organized than a data-dependent acquisition. It looks similar in some ways to a parallel reaction monitoring, but covering more of the MZ space than a parallel reaction monitoring would. So with a data-independent acquisition, let's say if we were to take just one of these chunks, so this is a precursor MZ value, say this is 600 to 650 MZ. At this one retention time, we take an MS-MS scan. We program the mass spectrometer to make a mass spectrum for all the precursors from 600 to 650, say. Let's say this particular peptide happens to fall in that scan. we would get our MS2. But because we are monitoring that window, that MZ window across all of retention time, we can stack each one of those MS2s. Each one of these rectangles is another MS2. If we stack all of those MS2s across retention time, and we looked down all of those, so looking kind of down through all of these blue boxes, we get the chromatogram. So now we can do everything chromatogram based for our quantification. This is super powerful compared to other DDA based approaches, because chromatogram based quantification tends to be as sensitive or more sensitive than even those isobaric labeling techniques. Plus, now you can do more than 16 samples. You can do hundreds, thousands of samples. And it's cheaper because you don't have to buy the TMT kit. So that was kind of two ways for us to think about DIA. You could think about it at a high level as just a way to balance number of samples, number of proteins you measure and how sensitive they are. Or if you're more of a mass spectrometrist, you can kind of think about it as that ion map. Kind of depends which way you're approaching mass spec proteomics, more from a biological question side or from more of like an analytical chemistry side. There's a lot of constraints and compromises. So one thing that Sue had kind of mentioned is you're limited by your hardware. So you can only make MS2 scans as fast as your hardware can go. And for mass spectrometers, we measure this in hertz, so like a scans per second. You're kind of capped by your hardware. Related to this, in order to get enough points across the peak for a chromatogram to be good, precise, accurate quantification, you need at least 6 to 8 measurements. So you can kind of see how those two play into each other. If you need at least 6 to 8 measurements, but your instrument can only make so many measurements a second, you have to do some math to figure out what the best way to run your instrument is. There are some compromises with DIA. The wider you make those MZ windows, the more interference comes into your quantification. We'll talk more about that later and like actually just show you what that means. And then finally, you can't measure across the entire range of the proteome. You have to choose where you want to measure. So we're going to talk a little bit more about what these mean. So what do different implementations of DIA look like? I want to give you just a kind of overview of different flavors of DIA that you might see as you're coming across this type of research. So the first, kind of technically parallel reaction monitoring, is a type of DIA. So you have your precursor scan, let's say from 400 to 1400 MZ, and then you're going to program your mass spectrometer to collect just very specific fragment ion scans. Then you'll take another precursor scan, and then your mass spectrometer will collect those fragment ion scans again. So you're collecting the same fragment ion scans across multiple retention times so you can build that chromatogram. So you might get a mass spectrum that looks something like this. It's pretty clean. This mass spectrum looks pretty clean to me because there's not a whole lot of peaks. We'll be showing you examples of DIA spectra that are very complex. If you were to widen out your parallel reaction monitoring, instead of doing each one of these individual, very specific fragment ion scans, if you go to the exact opposite of this, it would be like an MSE, an MS everything. And your mass spectra would look something like this. So this is now, instead of taking just one very specific fragment ion scan, what would it look like if you took a full fragment ion scan for everything at that particular retention time? You can see it starts to get a lot more crazy, right? Instead of having just one very specific fragment ion scan, now you can see there's lots and lots of peaks in here. So this is one way you could think of two extremes of data independent acquisition, right? So you could think of parallel reaction monitoring as like the most specific you'll ever get, and then MSE as the widest, least specific you will ever get. Either very, very narrow windows or very, very wide windows. So MSE, you might think, well, why don't we just do that? That seems easier than trying to figure out the precise Goldilocks fragment ion window width. it would be great because that's a great advantage, right? You get to quantify with your fragment ions using that chromatogram-based quantification. You would get very, very frequent scans because your instrument is just flipping through all of these fragment ion scans, and you don't have to compromise the range of MZ that you're measuring. See, the problem is you can't really analyze the data. You would have to try to come up with a computer algorithm to split that very complex spectra into its constituent peptide spectra. And that is very, very difficult computationally. So this approach is not typically seen too much now. There's a little bit of a resurgence in interest in building those computer algorithms to try to deconvolve these peptide signals or come up with a way to analyze this data. So it might be something you see in like a 5-10 years from now, breaking research kind of thing. But for now, anybody use this approach because it's just way too hard to try to analyze. So compromising between very specific PRM and very non-specific MSE, there are a couple choices. What about a PRM just slightly wider MZ windows than a PRM? This very narrow window DIA is the Pacific method. And you would tile very small fragment ion windows and continuously tile them across until you've covered your full MZ range. So this very narrow window approach gives you simple enough fragment ion scans that you can really basically use DDA analysis on it, but still get all the advantages of DIA for quantification. The disadvantage, though, is because you're using such narrow windows, almost no instrument is really fast enough to cover a whole proteome with this narrow window DIA. Maybe in the future, there'll be an instrument fast enough to do this. But for now, you would have to inject each one of your samples multiple times, multiple injection, in order to cover the full MZ range of a proteome using these super narrow windows. So super easy to analyze, but kind of a pain because you would have to do these multiple injections for each of your samples. Okay, so the next thing, okay, well, what if we make them slightly wider, but not quite as wide as MSE and looking at everything? So a wide window DIA, Swath, or DIA, they're the exact same thing. Swath is just a branded, like, trademark name for DIA. This is where pretty much everyone in the field is using their DIA these days. So it's a slightly wider scan, and you tile those across the full MZ range that you want to measure. Take your precursor scan and then tile them again. So now every cycle of your DIA, you're getting MS2s for every peptide across the MZ range that you're interested in looking at. There's some variations on how exactly to set this up, but this is generally what you'll see everyone in the DIA application method section doing, is some slight variation of this type of DIA. So there's a lot of benefits. You still get that quantification with fragment ions. You get your frequent scans, so you get enough points across the peak, but you're not quite as reliant on deconvolution as that MSE if you were to have super, super wide fragment ion scans. The disadvantage, you still have to pick what your precursor MZ range is going to be, and that is not necessarily the same for every type of sample, so that takes a little bit of method development to figure out exactly what range you need to do. And you still need to use special software to analyze this. You can't just use a DDA software for the most part. You have to use special DIA processing softwares. So advantages and disadvantages. So just to kind of reiterate, there's that super, super wide MSE version of DIA, and that spectra just looks super, super complex, right? If you compare that to these DIA Swath methods, on the bottom, these MS2 spectra look a lot more typical for what we would expect to see a peptide mass spectra. Not quite so many peaks, and you can kind of, for those of you that are kind of in proteomics, you can maybe a little bit more by eye start to see the spacing of amino acids in the MZ range. So MSE would be super great. The computational proteomics isn't quite there yet for MSE, but maybe in the future. For now, DIA, definitely the flavor du jour for doing these DIA experiments. So kind of, I think this is the most popular DIA windowing scheme that I tend to see on Orbitraps. So if you're on a TOF instrument, I don't know quite as much up to date with TOF instruments, but you take each one of these fragment scans, you take 25 of these fragment scans, and each one of them is 24 MZ wide, covering 400 to 1,000 MZ. You take another precursor scan, and then you start all over. Another 25 windows of 24 MZ width. So now you've covered everything. If you think back to that ion map, the goal is to try to be covering the darkest part of that and intensity across the strip of MZ values. So we mentioned on your instrument, some of your instrument, you're kind of capped by the hardware, how fast you can go. For a Q Exactive HF, it takes about 2.5 seconds to go from one fragment ion scan at 400 MZ to cycle through all the 25 windows, take a precursor scan, and then get to the next 400 MZ. So in your liquid chromatography gradient, you would have an MS2 scan, you would have a point across the peak every 2.5 seconds. Okay, so you have all these fragment ion scans. Let's take, for example, just one of those fragment ion scans at some particular retention time. At a given retention time, that fragment ion scan gives you an MS2, a mass spectrum. But remember, we're taking multiple of these measurements over time. So we have multiple MS2 spectra as the peptide is eluting off the column in your liquid chromatography. So retention time is moving on. And as the peptide elutes, we take one MS2, and then the peptide elutes some more. We take another MS2, peptide elutes some more. We take another MS2. So we're getting these points across the peak. So now we build our chromatogram across all of these individual MS2 values. We will see chromatograms, see peptides start from low intensity, build in intensity, and then go down. But we do have caveats when we're doing these DIA style experiments. The first is that the ions, this is more for like the mass spectrometry crowd. If you're coming more from biology background, this is a little in the weeds for you. You don't super have to understand the details of this, but for the mass spectrometrists, the ions aren't going to necessarily be centered, and your collision energy is not going to be optimized for each individual peptide. So for DIA, you have a fixed collision energy across your entire experiment. And the ions aren't going to be perfectly centered in this bucket of isolation values. So you might get some peptides that are kind of on the edge. There's almost always going to be multiple peptides in the window. And these edge effects, if you have a peptide that happens to be sitting kind of on the edge, the mass spectrometer, the quadrupoles, aren't perfect at transmitting ions. So towards the edge of the windows, you'll get decreased efficiency. So this is a good spot for me to stop and take some questions before we go to the next session. So Angel has a question. What is the resolution for this scan? The resolution you choose depends on your instrument. So we have a paper that I can put in the chat. I can have, I'll remind myself to put a paper in the chat that is, I'm the first author. It's MCP 2020, Acquiring and Analyzing DIA, blah, 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 blah. But it has a whole table for Orbitrap instruments that does the math for you and balances how wide the windows can be. And what resolution to use to get that speed, because if you increase the resolution, you're gonna take longer to get your points across the peak. Paul asked, do measurements for peptides that exist right at the MZ boundary suffer? Yep, exactly. That's that quadrupole transmission problem. We try to get around this, DIA researchers try to get around this by using what's called forbidden zones. So based on kind of average isotopic distributions of peptides, you can kind of theoretically calculate where there cannot possibly be a peptide ever. We try to place our edges of the windows where those forbidden zones are. So that's like a very deep in the weeds mass spectrometry answer. But yeah, those measurements don't end up suffering because we position those windows in the forbidden zones. Or you can. Not everybody does, but everybody should position their windows at those forbidden zones.
One thing we should point out is the forbidden zones are really for charge 2 and 3, 1,
Yes.
2, and 3. And if you go up to higher charge states, they work. They don't work as well. So.
Yep.
But, yeah.
And there's also caveats too. If you're doing something like phosphoproteomics, phosphorus makes a very different forbidden zone profile than the regular carbon, nitrogen, and oxygen. Very in the weeds answer, but I can throw some references in the chat, or I'll throw references in that document link on the May Institute website so that you can have them after this Zoom gets shut down. So in the DIA scheme, does the software stitch together the fragment scans? It doesn't need to stitch together the fragment scans to ID the peptides. A lot of DIA software will look directly at the fragment ion scan and analyze it from there. So you don't need to use the chromatogram to ID the peptide. You're still doing the identification, the detection at the MS2 level. Are there benefits to doing slightly overlapping windows for DIA? Absolutely. If you use slightly overlapping windows, if you're on a TOF, having the margins on your windows is good. If you're on an Orbitrap, do not use margins. Instead, do a full stagger, a 50% stagger. So that is also discussed in that MCP 2020 paper that I'll make sure I link in the documents. Jonathan Wilson, 6 to 8 precursor scans are required per sample per peptide. Yeah, so not the precursor scans aren't as important as the MS2 scans. Precursor scans, there are some people that make their DIA methods with no precursor scans. And depending on the software you use, you don't need precursor scans at all. You can just go straight MS2 fragmentation scans, and you want 6 to 8 of those scans so that you get 6 to 8 points across the peak in your chromatogram. And the fragments, it's everything in that, any peptide that falls in that 25 MZ wide window will get fragmented together. So you'll have a chimera spectra, you'll have multiple peptides in the same spectra. That's why it's kind of nice to use like a DIA software to be analyzing this because they're specifically designed to take that into account. So the point of overlapping window DIA, so depends what you mean. For margin overlap, the point is this transmission efficiency. By having the slight margin overlaps, you don't have that transmission efficiency problem. The other way around that is to just use the forbidden zones and put your window boundaries at the forbidden zones. For the point of a staggered window DIA, you can use computer algorithms, computational algorithms that will deconvolve the stagger so you get twice as narrow windows. So we can talk more about that if people are more curious about that. That's getting a little bit more into the weeds of a very, very specific flavor of this popular DIA scheme. Do you keep shifting the windows and sequential injections to cover the whole proteome? That is for that passive method, the very, very narrow window. You would, each injection, do a different MZ value. For this most popular DIA windowing scheme, you do not do sequential injections. Each sample is just injected one time, and you tile across this for every sample. So sequential injections, yes, each method, each sequential injection would have a different method for a Pacific style narrow DIA window method, but for this most popular style, this wider window, you don't do sequential. Where are you fragmenting the precursors? Same place you fragment them for PRM. So if you're on a tripod instrument, if you're on an Orbitrap, if you're on a TOF, you just fragment them all together and take the scan of the full kind of bucket of all the peptides that gets fragmented. Yeah, as far as using MSE, the software, as far as like getting good quant and identification, using MSE as a trademarked word for DIA, that's totally fine. Using MSE as the method of having every fragment ion going into the mass spectrometer at the same time, I don't recommend. Using MSE as just Waters branded DIA, that's totally fine. Use a method like this most popular windowing scheme, this wide window. That's totally fine. Okay, I think we're just about caught up with Q&A. So we started getting a little bit into this. So since the spectra are a little bit more complex, how do we analyze DIA experiments? There's a lot of options out there, so I wanted to put up maybe a couple considerations that you might have. We're not really going to talk too much about libraries, besides that sometimes you need them. We're not going to get too much into the weeds there. If you want to talk more about it, we can talk more about it afterwards. Does the software let you look at your data? And is the software free? Maybe these are like the top questions I get when people ask me. So of those, there's a lot more options than just the ones I'm listing here. But these are, I think, maybe some of the ones that I see the most. So Open Swath is a software, requires you have this library and it's free. It doesn't have a good way for you to interact with your data and visualize it. Same goes for Swath 2.0. That's kind of a commercial version. So it's not free. Skyline, which you're going to be doing tomorrow and Wednesday, does require you have some sort of library. We're not going to talk about different types of libraries, but it uses a library. You can interact with the data and it is free. Spectronaut, you can use a library or not, and you can interact with your data, but you have to pay license. DIA Umpire is another popular one, was maybe more popular 10 years ago than it is now, but it's also a free option for you. Encyclopedia, this is the one that I tend to use the most. It does require that you have some kind of library and is free, but you don't really have a good way of interacting with your data. And Diane, D-I-A-N-N, Diane is another very popular one right now that's free, but doesn't really give you a good option to visualize your data yet. So you'd have to pull that data into something else to visualize it. There's a lot of options. It doesn't matter so much what you pick, but one hill that I will die on is no matter what you use, you should look at your data. Don't just believe the matrix that the quantitative matrix that these software spit out. Don't just take them at face value. Go and look at your data. And this is why. So this is a screenshot from Skyline looking at a chromatogram. So retention time on the x-axis, the time the peptide is eluting off of the liquid chromatography column, and then the intensity that is measured by the mass spectrometer. Each one of the colors is a different transition for this one peptide. So transition is just the fragment. So the peptide, we break it in two parts, and that part is the fragment from the peptide. So each one of these different colors is a different peptide. This is an example of a pretty decent looking peptide. We're going to look at some more crazy ones with Brendan tomorrow and Wednesday. Just kind of orient you. So we looked at Skyline before, but this is just how this chromatogram is just one piece of Skyline. If you were to zoom out and look at the Skyline software itself, it looks like this. So on the left-hand side, you have all of the targets. You have to have these targets in the document before you do anything else. Skyline needs to know what it's going to look for, right? So this hierarchy goes protein at the top level, and then the peptide sequence, and then the MZ value for that peptide with a charge state. So the plus means it's a +2 charge state. And then all of the transitions underneath here with their MZ values. You see their +1s. So the chromatogram window, just a tip for you for later, if you happen to lose this, it's very easy to get the chromatogram window back. Here I'm showing just the MS2, so the fragment ions, because that's 99% of the time all that I care about. But you can also see precursor ions if you happen to care about those. There was a question from before about analyzing DDA data in Skyline. There is a Skyline tutorial for doing MS1 full scan filtering. Maybe I wouldn't like super highly recommend MS1 full scan filtering for your quantitative proteomics needs, but if you happen to have data that's DDA and you want to pull it into Skyline, you can look at it that way. And what we're going to be doing today, hands-on, is going a little bit more into what does it mean for a chromatogram to look good? Because as you're going through this May Institute, if you're sticking around and doing more of the statistics, garbage in, garbage out, right? There's no statistics in the world that will save a bad data set if the data set itself is bad. Can't massage biology into a bad data set. So some of the things that we're looking for in a good quantitative data set, the peptide signal should all co-elute. So you see this blue signal is not perfectly stacked with the red, orange, and purple signal. You want them all to be a perfect rainbow, all perfectly co-eluting. The peak shape, ideally, will be very symmetrical. This peak shape is not bad, to be honest, but it's not perfectly symmetrical. It has a little bit of a tail. Signal intensity, as you get into more complex data, you will start to see other places in the chromatogram that could be the peptide. These top three, you don't need to know anything else about your experiment besides having raw data from your mass spectrometer. If you're really looking to go deep, especially with DIA, you should have a library spectrum. Where that library comes from, there's a lot of options. We're not going to have time to go too deep into those, but feel free to ask more if you're curious. The correlation to your library spectrum is very, very helpful in knowing that you picked the right peptide. Similarly, the correlation to the retention time, whether that retention time is predicted or measured from a real library that you collected on your instrument, can be helpful in knowing that you picked the right peak. And then finally, if you are like super gold star A+ mass spectrometry proteomics experiment, you'll have a paired internal standard that has a heavy lysine or heavy arginine, and those two should perfectly co-elute. So that if you look for your heavy standard and your light standard should be right there, perfectly co-eluting with it. That one, not so common with DIA, but very common with DIA, maybe even required for a lot of DIA softwares, is the correlation to a library spectrum and correlation to a retention time. So there was discussion about how good the data looked before. We're definitely going to be looking at much messier data now. But this is what I mean by interferences and why maybe the commenter was saying like, wow, this data looks perfect, but what about real data? This is more maybe what you'll see in real data. So where exactly is the peptide? So using these criteria, and we're looking at this, and this is what we're going to be doing hands-on, looking at this data, where exactly is the right peptide. Especially on the left, you see there's in the orange Y4 signal, there's a huge interference, and it almost makes it hard to tell that right here, a little after 13.5 minutes, there's a beautiful stack of all the other fragment ions in a very symmetrical, pretty looking chromatogram. But there's this giant orange signal interference in that. Similarly, another type of interference that you might see in your data, here's another example, just about at 18.5 minutes with an orange, a blue, a purple, and a red fragment ion all stacked and looking pretty symmetrical, except for this giant interference in the Y6 signal. So this is kind of what we're talking about when we're talking about messy data, and in DIA, you're more likely to see these interferences than if you were to do an SRM, a targeted SRM, MRM, PRM style experiment. Skyline is super good at detecting clean peak groups. So this is an example of a data set where it's very, very obvious where the peptide is, and Skyline picked it correctly. As data gets messier, it becomes more difficult to pick the right peak. And this is why, and it's not even like a Skyline specific issue. This is every software out there. And this is why I have to emphasize so strongly, look at your data. Don't just trust the quantitative matrix that gets spit out from Spectronaut, spits out from Diane, whatever you're using for DIA. Look at that data. I use Skyline to always look at that, but look at that data and you might find it has incorrectly identified something. And whether you're doing your own mass spectrometry for your own research or whether you're like core facility working for a collaborator, nobody wants to waste years chasing down something that ended up being just a misprocessed signal. So double check before you publish, before you give your collaborator any answer, double check that it's real and that the signal is convincing. Skyline has a lot of tools for you that we're going to use. And I only have a couple more slides before we'll do the hands-on. So I'll definitely get to the questions too. But Skyline has some tools that we're going to use that make it very helpful to quickly curate data, even across pretty large data sets. So here is a retention time screenshot from Skyline, where across the x-axis going left to right, these are all the different sample replicates for this experiment. So these are each individual sample run on mass spectrometer and the data pulled into Skyline. And then on the y-axis, we're looking at retention time. In the box is the start of the chromatogram, the start of that peak, and then the end of that peak. And if you look across, there's a little bit of wiggle room for all of these samples. Retention time isn't perfectly reproducible across different samples because each sample has a very slightly different matrix, and even slight differences in matrix will change the retention time just a little bit. That said, there is one sample that is very clearly an outlier. So this would be an example, as I'm looking at a data set, I would say, aha, this looks like it might be a mispicked peak. So I could click on that, look at that chromatogram and ask myself, do I believe that the software picked the right peak here? Or do I need to manually curate this? And I, as the expert, know that the peak is actually over here by 31 minutes and not at 33.5. There's other tools in Skyline that will help you look at this. So this is a similar style of data curation tool where we have the replicates going across the left to right x-axis, and then the peak area percentage. So these are all of those different fragment ions. They should all, across all of the samples, they should go in roughly the same order. So if red is always the tallest, and then teal, and then green, it should always go in that order. So when you see across these samples, red is almost always the largest percentage, you see that there's an outlier right there, where that chromatogram clearly didn't follow that same fragment ion order. The rainbow was a mix of colors, and it should not have, the colors in the rainbow should not have gotten mixed up. So between the retention time and between this peak area percentage, this is a signal to me as the expert to go back and manually curate this data before I try to push it downstream for statistical analysis. So that was pretty much everything I wanted to cover before we do the hands-on. Active areas of research, in case people have questions about this, the best practices for doing PTMs by DIA. This is still like a little bit of like maybe not a super figured out field. Predicting retention time and fragmentation for in silico spectral libraries. We're not really talking too much about that, but super popular way to do DIA is with these in silico spectral libraries instead of doing the DDA yourself. As data sets get bigger, your computer might not be able to process them. You might run into this tomorrow or Wednesday if you're operating on a laptop, for example. So people are working on cloud-based resources so that we can process larger data sets. Then finally, if you're coming from the DDA world, you might be familiar with FDR and decoys. This is a pretty big active area of research for DIA because the way we make decoys for DDA doesn't work great for how we do DIA. So doing proper decoy generation and proper false discovery rate is something to always think about with DIA and a little bit of bleeding edge in the field. So again, this was a lot of information. Really the only 4 things that I hope you can do now. Key differences between DIA and some of the other acquisition methods. that there's different flavors of doing DIA. It's not quite as cookie cutter as DDA or PRM. The caveats of DIA and the data analysis, main takeaway there, look at your data. And then finally, we're going to look at some actual real life data. So while I get spun up to do that, we will go through questions. It's a question about Skyline for dose-dependent analysis. I don't think there's any statistical analyses in Skyline for dose dependency, like a dose response curve or anything. So nothing inherently in Skyline for that. But if you're sticking around for the rest of MS Stats, that would be a good place to ask them. How do you do manual curation in Skyline? That is literally the hands-on that we are going to spend the rest of the afternoon doing. Perfect. And then a couple much longer questions in here that I will try to get to. It looks like Brendan is getting one of the transition ones. So I'll get to Mark's questions, questions and comments on fragmentation and DIA. If you are fragmenting in the ion trap that uses resonance ionization, which is automatically optimized. We do not do automatic optimization for DIA. Nope, this is all NCE, and you have a fixed NCE across all of your scans. This is also true in QTOF too. So you do not do any sort of collision energy optimization for DIA. You pick one collision energy and you fix it across all of your fragment ion scans. So the issue comes, and this is getting a little bit into the weeds, the issue with this fragmentation energy, this collision energy, comes with the fact that if you have a DDA spectral library and you did not acquire that DDA spectral library with a fixed collision energy, then the comparison between your DDA spectral library and your DIA acquired fragmentation will not look the same. The more those don't look the same, the more dissimilar those look, the worse the dot product will be when you compare them. And the worse the software that's trying to analyze your data, the worse score it will give that peptide. So if you are going to collect a DDA spectral library, collect it with one fixed normalized collision energy that matches the fixed collision energy that you're collecting your DIA with. So it's kind of that question of getting a library that accurately reflects the real samples that you're running with not only the samples, exact samples that you're running, but also the exact instrumentation you're running as well. This is not necessarily just true for DIA. If you're doing PRM and you happen to have different collision energies, or you're using a different instrument to acquire your DDA, then you're going to go on like your triple quad or something. That mismatch between the library spectra and the acquired spectra on your instrument can give you a low dot product when it was just a collision energy issue. So that was like very in the weeds, but we can go back into that. Question about what's challenging doing DIA for measuring PTMs. Part of it is the issue of isomers. Typically, there are co-eluding isomers at the same retention time, and how you deconvolve that signal can be very difficult. Another issue is libraries. If you're using in silico predicted libraries, there's not great ways of doing that right now. Maybe phospho is like fairly well figured out, but for other types of PTMs, it's not quite as well figured out. It also becomes difficult if you're looking at small PTMs, like an acetyl or a methyl. Those can fall in the same window as their unmodified form. And now are you detecting and quantifying the modified or the unmodified form? There's big, huge area of active research there. But at the end of the day, it's definitely, maybe I would be a little cautious doing DIA for measuring PTMs unless I had a paired internal standard so I could be absolutely certain that the quantification was correct. We have a couple more questions. See if I could do a quick one. Do you have to generate your own DIA libraries? A lot of people are using predicted libraries these days. I don't know very many people who are now doing paired libraries, unless they're doing the approach that I tend to use, which is the chromatogram library-based approach, which does generate a DIA library. Most people are using these predicted spectral libraries these days. They're pretty cool. Can DIA incorporate ion mobility data? This one may be a better question for Brendan, but Skyline can incorporate ion mobility data. I don't know if that is incorporated in the Skyline DIA analysis in the way that you happen to be thinking, but ask that again tomorrow or Wednesday.
Yeah, I'm here.
There you go.
Yeah, Brendan. Yeah, definitely. I mean, there's actually a whole tutorial on, if you're thinking timsTOF, there's a whole tutorial on DIA process. You know, we have definitely worked with FAME's data and believe that, you know, if somebody, I'm not sure anybody's given us a FAME's DIA file yet, but I think that Skyline should be capable of doing that. Yeah, so we've been working with ion mobility data for a pretty long time, and Skyline has lots of functions and features. And so, yeah, if you get started with some aspect of DIA with ion mobility and you don't feel like it works well in Skyline, just contact us. We put a lot of
nice. Yeah, I know there's some really cool like scanning swath, like sonar style, where it like slides the window across. I don't know any software that can handle that particularly well, but that's a pretty cool approach.
Our understanding is that Cylac on their side is doing the work to make it look like normal swath to us, so we just process it like normal swath. But yes, they're sliding the windows and doing something more complex, demultiplexing, and then, but we don't ever see that data.
Yeah, I personally have never interacted with that data. It's cool. I don't have access to it. Okay, so we're going to get into some of the hands-on. So what we're going to do is open up making sure I know where all of my materials are. Okay, so you are welcome to follow along with me on your own computer, or feel free to just watch me for now. And these are going to be recorded, so you could watch me this time and then do it on your own. Or if you feel good and confident, grab the PDF and just go to town and you can just listen to me in the background and let us know if you get lost. So in the linked tutorial materials, there's this group studies folder. If you unzip that and you dig deep down, you will find this rat_plasma.sky.zip folder. You could unzip this here and open up the Skyline file, or as you boot up Skyline on this start page, there's an open file. If you navigate to where that .sky.zip is, Skyline will unzip it for you. So you don't necessarily have to unzip the .sky.zip. You can just open up directly in Skyline. So I'm opening up that, getting everything opened up for me. You should have a screen that looks something like this. Maybe the most important thing you will learn this whole week is how to change the color scheme in Skyline. My colors of my fragment ions might be different than yours. To change that, on the top left, it's tools, options, display and you have all these color schemes. So I'll switch mine back to classic for now, so that we're all on the same page. But this is probably the most important thing, so that when you make your posters with Skyline screenshots, you can have your pretty color coordinated poster scheme. A little bit of orientation. On the left-hand side, we have all of those targets. So remember, protein level at the top. If you hover the mouse over the protein, it'll give you kind of all the FASTA info. Then the next level down, we have the peptide. And then if you click on the plus sign next to the peptide, that will show you the precursor MZ value for that charge state. And then the fragment ions that were chosen and refined for this project. So we're walking into this project. The peptides, the target list has already been chosen. They've already acquired all of their data. And now we are kind of the data analyst, and we need to go through and analyze all of the data. So another kind of piece of orientation down on the bottom right, below the replicate screen, there's a 1 out of 48 proteins, 1 out of 125 peptides, precursors, transitions. This is kind of a nice way to get a quick overview of what's in your Skyline document. So some more orientation. You may notice over in the target window, for each peptide at the peptide sequence, there is a little P with what kind of almost looks like an antibody underneath it. Some peptides only have the P with the little like red antibody. Some of them also have a couple of sticks in their icon. If you click on one of those peptides that has the little sticks, you can click on any of them. For me, I'm going to click on peptide LGGEE, blah, 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 blah. That little icon means that there is a library spectra for it. So with Sue, we just looked at some library spectra. There's a couple different ways that you can view this. I'll show you the way I typically use, which is at the top, at the menu, select View, Spectral Libraries. So now you can look at all of the spectra in your document. And Sue was kind of showing us how typically you would want spectra that kind of span across the MZ range. That means there's like lots of fragmentation. You want good fragmentation for your peptides across everything. So you can look at all of your fragment ion there. You can also go to the top right, go View, Library Match, and that will open up a window for the peptide that you're on. And now as you click or as you use your arrow keys to select different peptides, it will show you the matching spectra for the peptides that have spectra. This first one does not have a spectra for whatever reason. Either this peptide actually isn't seen with DDA, or maybe the sample that the DDA spectral library was collected on, maybe this peptide wasn't present in that sample. For whatever reason, there's no spectral information for that first one, but the rest of them, you can kind of see each one of them. So these samples, already some of these came preloaded. Oh, I will double check the Q&A. Sorry, I had navigated away. A couple questions. How are peptide spectral matches actually matched? So the spectral library has already been searched with another software. You could either use within Skyline, MS Amanda is now available to search directly in Skyline and build a spectral library from that DDA, or you could use whatever your software of choice, Proteome Discoverer, Comet, Mascot, Sequest, whatever you happen to use, MaxQuant, and then you can build spectral libraries in Skyline. with settings, peptide settings, library. You have these spectral library options. We'll try to drop some links to step-by-step tutorials in the chat. So now that peptide sequence is already associated with that spectral match. So this peptide sequence in Skyline has that knowledge, that prior knowledge of what the spectra should look like. So it doesn't have anything to do with the MS2 peaks. It doesn't have anything to do with points across the peak. It's just DDA. So was your DDA search able to find that peptide in your spectra? For single cell proteomic data, I don't have any experience with single cell proteomics or doing DIA with that. The spectral library, all I can really advise is the spectral library should be as representative of your real samples as possible, because the way fragmentation and matrix effects kind of act together with retention time, you should try to have it be, if your real samples are like nanogram levels of peptide, I would make my spectral library with nanogram levels of peptide. And now in Skyline, if you use inserted protein peptide instead of library, can you still view the spectra? Only if you already have the library set up. You could insert a peptide and it would associate that. But if you don't have the library set up, Skyline will not like make up a spectra for you. Okay, so it looks like we're mostly caught up. Hopefully everyone's just kind of like tabbing through the data, getting familiar with it. So we are going to just practice pulling in some data. So if you go to File, Import, Results, File, Import, Results, you'll get this Import Results window. So this is, I'm done acquiring a raw file or a WIF file on my mass spectrometer. You do not need to do any kind of conversion. Skyline will natively read across all the different vendor formats. You can even pull in different vendor formats into the same Skyline document. That was kind of like one of the reasons Skyline first was invented and like came to be, was there was a big giant study and people were running different types of instruments, different vendor instruments, and there was no good way to analyze all the data together. So in this import results window, we're going to keep the radio button at add single injection replicates and files and hit OK. Now it's going to pull up a file browser window. Navigate to wherever your tutorial files are and go to the raw folder and click on just the first raw file. That's this D_102_rep1. Then you can hold down the Shift key and grab the last one. So there's other ways you can do this. You can click and drag in the window. If you were a glutton for punishment, you can click each one with Control. I typically do the Shift and then hit Open. And now Skyline shows you what some instructors like to call Skyline TV, which is showing you all the chromatograms as they import. So Skyline, the way it's working kind of under the hood, all of these MZ values that you told Skyline to look for, it's going to look across all of your data for the scans that match those MZ values across the retention time that you set up in your settings. So if you're doing DIA, probably you would not choose to import everything across the full retention time, but maybe you are. This data is not DIA or DDA data. This data is SRM. So this is like a targeted experiment. Tomorrow you will be working with DIA data, and I think it has a DDA paired spectral library. I think this data is not PRM. I think it's SRM. I can check. Yep, SRM, selected reaction monitoring data. So there's more questions in the chat while my data is finishing loading in. How do you get a peptide match when there is no fragment spectrum in the library? So Skyline is actually doing its own kind of searching and scoring under the hood. It's taking all those MZ values and it's creating very similar score criteria from what DDA search engines do to find where the peptide is in your mass spec data. So Skyline does not require that you have a library in order to analyze data, especially this kind of data, this kind of SRM style data. It's nice to have because Skyline can use that as an additional scoring measure, and you can use it as an additional kind of sanity check. So as you're selecting peaks, if you don't have an internal standard, you don't have a heavy peptide, and the peak is consistent across 3 injections, is that correct? That would indicate to me that probably it is correct. You could also look for other potential peaks in your full chromatogram. Other peaks might look similar across your chromatogram. We're going to hopefully show you how to do that in just a minute.
So one thing, so there's been a lot of like, what if you don't have a library, but and the data that Lindsay is showing, this data set at the time, we used just the public libraries and found that only about 50% of the peptides we were interested in targeting had library spectra. And so we started this experiment just targeting all tryptic peptides, and then these are the ones we thought we could detect. But we now, since then, we have PROSIT integrated, and you can actually, one of the things I now frequently demo here is you can see which peptides have spectra, and there are these little scratch marks on their icons, these little, like look like a little spectrum right down at the lower right corner of the icon. Yeah, Lindsay's hovering the cursor over it. So, but you could get all of them to have something by going and building a PROSIT library. And I frequently do that when working with this data now. It's just build a PROSIT library, and then you have something where you can look at a .p value, and we've proven that's a really great option if you don't have a spectral library.
Yeah, totally agree. I do that a lot too when I'm building a PRM. Instead of making a DDA library or something, I'll just throw it into PROSIT. So now we have kind of all of our data. I wanted to show you, there was a question last time about how to arrange the windows. So I'm going to close my library match. I'm going to close my retention time. I'm going to close my peak area so that I just have the chromatograms. And I just want to show you kind of how you can manipulate what Skyline looks like, because that was a question from last time. So first, you can click and drag and lock, like kind of dock any of these windows wherever you want. So for example, now I'm going to click on targets. I'm click and hold on targets. And you see now I have this like blue window. I'm click and holding, and I can drag that wherever I want. You see, as I'm dragging it, some of these kind of windows are popping up and give me the option to dock at this location. So over here on the far right, if I finally release that click and drag, now my target window is docked on the right-hand side. I'm going to drag my target window back to the left-hand side because that feels very weird to have the targets on the right-hand side. I never see anybody do that, but maybe that somebody is you. So now we are going to pull up the retention times window. So view, retention time, replicate comparison. And now we get that retention time window back. So let's click and drag retention time to the bottom. And then let's view peak areas, replicate comparison, view peak areas, replicate comparison. And that one already came up for me, docked to the bottom right where I was going to put it anyway. But you can click and drag and get that docked right next to retention times on the bottom. Okay. Now your window should look maybe something like this. Now for our chromatograms, we're going to do view, arrange graphs, grouped. So view, arrange graphs, grouped. We're going to group these in 3. We are going to select the radio button, distribute graphs among groups. Under sort order, we'll have document. And all of this is in the PDF. I'm on the PDF page 8 if you were following along. So it should look something like this. We hit OK. For me, I had it kind of already arranged that way, so it didn't really change too much. Looks like maybe we're still doing good. So you should have something that looks generally like this. Now let's start actually looking at some peptides. So the first peptide I want to look at is GILAA. So you can find, just like in a Word document, you can Control F and enter in a peptide that you want to find. You don't have to have the full thing typed in. Oh. And my document is not finding this peptide for some reason. That's okay. I can find another peptide. So when we're looking through a document and we want to analyze it, I had told you before that we were kind of looking for these peptides that might not follow the trend in retention time or the trend in peak area. So you can tab up and down through different peptides using your arrow keys up and down. That's typically what I do. Or you could use your mouse to click on them. So we find some peptide. I'm on peptide AFMD. To find that peptide, AFMD. Find next, and it should take you to that peptide. You could do edit, find, and type in that peptide, find next to find that peptide. You see this particular peptide, it definitely looks like maybe there's a lot of dirty signal in here, right? Like these look pretty awful. If I look across different measurements of this peptide, I can click on their different retention time boxes and it'll take me right up in the graph to where that peptide is. And this looks super messy. This is an example of a peptide that I would not feel very confident in. Where is this peptide? So if I click on some of these, one thing you can do is take your mouse, hover where it says retention time in a graph. You could either use a scroll wheel to zoom out and see the full measurement for this peptide. See how messy this data is? So the first data set we were just looking at earlier, that plasma data set was super, super clean, right? It was so obvious where the chromatogram was. And now we're getting into data that is a lot less clean to look at. So over the next kind of 5-10 minutes, I'm just going to show you how I kind of tab around and might check some of these things. So I might kind of peak area. I might normalize. If I right click in peak area and I normalize to total, now I get that peak area percentage that I was telling you about before. So remember I was saying retention time might not be perfectly reproducible across different samples because of matrix effects. the peak area percentage can give us another kind of clue whether or not we've picked the right peak across all of the samples. So see, even though retention time for this peptide is bouncing around between, 18 minutes to 19 minutes, the peak area percentage is very reproducible across this. So we can look across a bunch of these peptides, and some of these chromatograms, this example on ENLSPP, this chromatogram looks great. If there was a chromatogram that did not look great, so we can go back to some of these guys down here that looked awful. So this peptide AFMD just looks awful. The retention time is all over the place and the peak area percentage is all over the place. So trying to decide which of these peptides is correct. It doesn't have a library spectra. How do you know which one is which? You might not, just period, know which one is which. But if I want to assume that this peptide should be around 18 minutes, what I want to do is integrate the area at 18 minutes. So these examples of outliers up at 21-ish minutes, those I would click. I would click that peptide. I would find where the window is, and I would zoom to the 18 minutes, and then underneath the x-axis, I would click and drag where that 18-minute is. This is so that I'm integrating the area. The quantitation is going to be with all of this same mess as all of my other samples. So I would try to align all of the retention times to about that 18-minute mark. Some of these are maybe too early, some of these are maybe too late. But I would try to integrate the same area for everything for this super messy peptide. Some of these are very obvious and they look great and you don't have to do any manual curation. So if we just keep tabbing down and just look across these, a lot of them look awesome, right? And before we do our statistical analysis, we would just want to find any of these that look maybe a little messy. And see if we can make sure that their integration matches. That's kind of the data processing that I might do. Oh, there was a question, how do you plot the area percentage on the y-axis? Right click in the peak area windows, normalize to total, and it'll give you the peak area percentage. The dot product does not include retention time. The dot product, and I can drop a link in the document, a link from the Skyline support board that describes exactly the various different dot products that Skyline will calculate for you. It's just a factor of the library spectrum match. If you want to look at retention time variability, if you right click in the retention times graph, and then go up to the top opt-in graph, you can look at regression, score to run. And this gives you the SSR calcs. This is the predicted retention time across all of the different peptides in your document. You can also click on these. All of these graphs are also interactive across Skyline. And you'll see some examples here of you click on some of these points and it'll bring you right to where the peptide is or where the peptide should be. So this is another example of curating your data. So here, this particular peptide, you can see in this middle graph that I have, so I'm on peptide IFPENNIK. This peptide got truncated. So this would be another example where I would manually want to curate this peptide and take everything from the truncated form of that peptide. Maybe this is a peptide because it's truncated. All of my statistical analyses downstream are going to be messed up because I don't get the full peak. So maybe this is an example where I wouldn't want to push this into a downstream statistical analysis because it's clear that the measurement has an error. This peptide has a measurement error. If I try to push this downstream for statistical analyses, then I'm going to kind of waste some of my multiple hypothesis correction that you'll be learning about later. But I'm going to test this peptide, and I know there's going to be this high variability because it's the measurement itself has a problem. So this is why with DIA, with anything, I would flip through every single peptide that I would want to follow up on and convince myself that it's real. So at the scale of DIA, maybe that's not practical. In this document, we only have 125 peptides. So that's reasonable to spend, you know, an afternoon with a beer, going through your Skyline project and curating your data, looking for all of these issues with the data. For example, this mispicked peak right here on peptide DYVSQ. This peptide, it's clear in this file that the right peak was not selected. It's another one of these truncated.
Lindsay, we have a hand up.
Yes.
Lee Foster.
I can.
Oh, sorry. I didn't realize there was another host that could unmute. I unmuted you again. You should be good.
Yeah. Since you promoted me, I have the power to unmute myself.
Yes.
So I missed the part where you changed your retention times graph to score to run regression. Could you just show me how you did that?
Yep. So right click in the retention time graph.
Mm-hmm.
And then in the pop-up, the top option graph. And then regression, score to run.
Okay, that's very cool. And I didn't know that you could do that. The other question that I had is when you have your peak area percentage and you've got it normalized, you know, it's very easy to tell when you pick the wrong peak. But I see you doing some kind of wizardry where you're able to eliminate those peaks that are not correct, and you're able to get all of the samples to align at the correct retention time. So how are you doing it where the ones that are wrong, you're just aligning all of them to the correct retention time, so it's so easy to find that peak?
Yeah, so maybe there's a couple things that I have enabled. So in the chromatogram window, if you right click in the chromatogram window, any one of them, it doesn't matter, you should get a pop-up that down towards the middle, there's an option synchronize zooming. If you click that and select that, now all of your windows will zoom at the same kind of rate. They'll all be scaled the same.
Okay,
that is a piece of wizardry. This is why I try to do a little bit more unstructured Skyline at the end.
Mm-hmm.
Just be careful on that one. So Lindsay, right click in there again in the chromatogram. I think you have the y-axis auto scale turned on for best peak. So the y-axis isn't changing when you do that synchronized zooming. You have to turn off the auto scale for the X and Y in order for it to be like everything adjusting properly at the same time. Like everything, both dimensions.
Yeah, my y-axis scale is not the same between these. So yeah, that is. If you hover your mouse and zoom on the y-axis, you'll see the intensities now are scaled. If that ever happens and you want to reset it, I always just go to another peptide and then click back and it resets. But here's another kind of example where as I'm curating my data, nothing got picked in this one. So I would just click and drag. So this is not maybe feasible for a targeted experiment. This is totally feasible. And if you're not doing this, shame on you. You should do this. For a DIA level experiment where you have, you know, tens of thousands, hundreds of thousands of peptides, not feasible. In that scenario, maybe you would do your data processing, you would do your analysis, and now you have your list of maybe a couple hundred significant peptides or proteins. Those I would put in Skyline, no matter what DIA data processing you're using, whether you're starting in Skyline for your DIA data processing, or if you're using Diane, Encyclopedia, Spectronaut, whatever you're using, once you have your list of significant hits, put those in Skyline and convince yourself that they're real and that the software that you had used had actually picked and processed the right peaks. So hopefully this gave you kind of a little bit of a deeper look into using Skyline and set you up for the DIA day tomorrow and Wednesday. I think we are supposed to be done now. So I will stick around a little bit, but thanks for joining us this first day of the Northeastern University's May Institute for Computational Proteomics. We'll stick around if you have some questions. Check those documents on the course website for any updates or references. We'll try to make sure that all that information gets in there. But thanks for joining us today, and good luck with the rest of the week. We'll try to answer any last questions you have. Thanks, everyone.
So maybe I'll just ask one last question.
Yeah.
So I mentioned in the Q&A that most of the time what I'm using is like a DDA to PRM workflow. And one thing that I always struggle with, and it confuses me every time, is the importing of the library and how you keep it separate from the actual data set that you're using, and you're just trying to reference that library. So could you just kind of elaborate to that workflow, because I'm still trying to get it straight in my head.
Yeah, so you're having trouble, do you mean like you'll get your spectral library in and then it'll try to like add every single peptide that you detected in your spectral library to the document, and you don't want that?
No, I've got a pretty good workflow for designating which peptides I want to look at and which transitions I want to look at, but it's just confusing for me to try to put the library in there, and then Skyline is trying to analyze my library, and I don't want to do that. I want to apply the library to get the right spectral matches on my PRM data set, and I get confused.
Yeah, so it should, if you do settings, peptide settings, and go to the library, and you already have your spectral library, you should be able to do edit list and add. And I'm trying to find where the tutorial is that I usually use for that.
Yeah, if you could just point me to a tutorial, that would be really helpful.
Yeah. I think it's in this one. I'll put it in the chat here, but I'm also going to put it in the document. So that everyone has it. Building spectral libraries. Skyline. Yeah, that's typically what I do. And it looks like we got everyone else in the Q&A. I will try to drop as many of these links as I can remember into the document for the course, and yeah.
So Lee, when you're importing or building a spectral library, if you import it, it should already have this .blib file extension on it, in which case Skyline isn't really doing much other than like opening it up. If you're generating or building a Skyline a spectral library, then you're going to be pointing to search results. That's Proteome Discover is going to be .pd results, or might be an MZXML or some kind of other result file, in which case then it will build it. And as it's bringing in the data, you will see it sort of what looks like analyzing as it's importing and opening it up. It's not really changing anything. All that it's doing is like building these spectra and labeling them for you and then matching it to what's in your peptide list. So there isn't really an analysis that Skyline's doing with it. It's more of just linking up what results are present and then what's in your Skyline file. If it's doing something different, I don't know what that would be.
Yeah, and I will say, if you do have, it happens to be like a specific issue with your Skyline document or your spectral library, the Skyline support board, I just dropped a link in the chat, and I already had it in the document. The guys are super responsive. They'll get back to you very, very quickly. They can help you out if it's like something weird with how your Skyline is exactly set up.
Yeah, thank you very much. I had another question. You kind of piqued my interest when you said that you use mostly these synthetic libraries. All the stuff that I've done has been experimentally derived. So do you have any experience comparing those two sets of libraries? And what would you say are the caveats of doing it completely synthetically?
Yeah, so I will say I've been very impressed with the predicted spectral libraries. They, in general, perform just as well or better than DDA.
Yeah, I did a presentation. You can find it on the Skyline website. I can post it into the. I presented at ASMS a couple of years ago, comparing PROSIT to empirical spectrum, even for a QTOF, triple TOF, Cylac triple TOF, and also for a Q-Orbi. And yeah, the spectra worked really well. It was impressive.
Yeah, I do know when I was looking at like the synthetic spectra, this was quite a few years ago, and I know there's been a lot of progress in this particular area, and I've heard that it's very good. I just haven't tried it myself. So, I mean, I guess I'm just wondering what the limitations of it are. Like, one thing that I find is that you have your theoretical peptide and you use it like as an internal standard, and then you learn that it either doesn't ionize well, or that the linearity is poor, or the LLOQ is poor, and you will only derive that experimentally. Is that something that they've gotten better at, or is this something that—
No. Any of those analytical figures of merit, you're not going to get from a predicted spectra. The predicted spectra will literally just be a spectra. It won't tell you anything else about the peptide, like linearity or LLOQ or ULOQ or reproducibility. It won't necessarily tell you anything like that. It will literally just give you a dot product for how well your acquired spectra matches the predicted spectra. So definitely not. Definitely hasn't gotten as good as that.
They're adding models, but I haven't heard. They're adding models like the PROSIT team has told me about some new models. We'll probably try to integrate this summer, but they tend to be for like charge state prediction and still like things like and whether or not a peptide is likely to even just show up on a mass spectrometer, but they haven't gotten to like predicting whether or not the peptide is going to be linear. And we may never, we'll see. Okay, so here is I just put in chat a link to the presentation where I talk about how PROSIT for the spectrum model and the retention time model, how it does compared with empirical data.
And I added that link in the document for this session.
Okay. All right.
Yeah, thank you very much. Appreciate it.
Nice job, Sue and Lindsay. Thanks.
Yeah. Yep, and Ivan, I added that PROSIT video to the document, so you should be good. NIST peptide libraries, you may have noticed in Skyline, if you go to settings, peptide settings, spectral library, there's a direct link straight to the NIST peptide libraries. So you can definitely use pre-configured, like downloaded spectral libraries from Peptide Atlas, from NIST, anywhere you can find a spectral library, you can get those into Skyline and use those. With the caveat that if it's not your exact instrument, with your exact instrument settings, you might have that issue with the dot product and the spectral library not matching your acquired spectra. Comments related to PROSIT generated libraries do also apply to Spectronaut or Diane library free. And this is just another one of those things where, like, no matter what you're using, Spectronaut, Diane, Skyline, whatever, look at the data before you send some poor grad student off on a wild goose chase to characterize some protein that ended up not being real mass spec signal. Just check the data. I think we're about 10 minutes over now, so I think I'm going to shut down this session, but join us again tomorrow, same time, same place. Brendan will introduce you to getting more in depth with DIA and data analysis using Skyline. So hopefully it won't be quite so much of a crash course in Skyline after seeing it a little bit today. Thanks everyone for joining us.
Thanks, everyone.